but it's down regulation. I don't know, there is some kind of cancer that show up regulation of NR2F6 in immunological system. But I cannot see some connection between this condition with skeletal muscle metabolism. In aging, we also look at the effect of NR2F6, and also it is repressed during age. Yeah, I have some finds I did not bring here, but it was a big surprise for us.
I think the most interesting thing is, and probably will be your next step, is try to understand how this nuclear receptor is regulated.
Yeah.
And I see two kinds of possibilities in there. You see, one is have to take a look in the promoter of this nuclear receptor to see what binds in there and regulates the amount of the receptor. And the second possibility is the ligand binding domain in the receptor. What is like that? Is you have an idea in the structure of that? What could bind in that?
No. We at this time we are studying the structure of NR2F6. We try to see if there is some kind of pocket, because nobody knows if there is pocket in NR2F6. Maybe there is no pocket to bind the ligand. The fact is, it is orphan receptor. We don't know how it is regulated. And then you try to do that, and if you find some possibility to bind something there, our hypothesis is if you find some antagonist molecule, maybe it can have a good effect on muscle metabolism. Yes, you have a look in complete sequence, and close to promoter, we found binding for NR2F6 in UCP3 and PGC1R. Okay, the promoter of my receptor. Yes, you have seen the promoter whole DNA sequence of NR2F6. And then you have a list of players that can be associated with regulation, induction, or repression of NR2F6. I cannot remind the names, but you have a list of, at least of 10, 12 proteins that can be associated with its regulation. It's a good question also, William. Thank you.
On the same subject, yeah. In your condition, do you know if there was some study of the 3D structure of the DNA? Just to see if there is some regulation by loop or DNA, spatial organization of the DNA?
No, I did not find some study in this way, but I think that NR2F6, as it seems to be a repressor of transcription, it might be possible. I think so.
Is there some additional question? Yes.
Thank you for your talk. Please, have you seen any change in satellite cells? Because like in cancer patients with cachexia, we saw an increase in PAX7 and MIF5, but a decrease in myogenin and myogene. So like it's kind of, they are trying to regenerate because of the inflammation, but a decreasing maybe in the differentiation.
No, we did not see. We just work with stem cells from skeletal muscle, from mice and humans in this work. And also we work with C2, C12 cells. But it's a good point that could be investigated.
Well, we will move on. Thank you, Leonardo.
Thank you.
So I will introduce Thiago Figueira. He got his PhD at the medical school of the University of Campinas. He's an associate professor at the School of Physiological Education and Sport in Ribeirão Preto. And his research interests lie at the interaction of mitochondria, bioenergetic metabolism, and exercise of physiology. So welcome and give a nice talk. You have the microphone.
Thank you for the kind introduction. First of all, I have to thank the organizing committee, particularly Professor Edesio, for inviting me. And it has been a pleasure to be among the speakers in this wonderful meeting. I'm going to talk here about some data we have got over the last years, which is related to a mitochondrial protein, an enzyme named nicotinamide nucleotide transhydrogenase, NNT, and we have found that it might be important for skeletal muscle aging. It's not changing. Nope. Okay, I will follow this outline here. First, I'm gonna show some background that for the purpose of everyone to understand what is NNT and its role in redox homeostasis. It's important to differentiate the structure and the function of these two nucleotides. On the left, we have NAD, and on the right, the NADP. And the only difference between the two is the presence of this phosphate group in here, but the function is a lot different between them. And on the left, we have NAD, which is more related to energy transduction from redox reaction to the respiratory chain. And on the right, NADP has several functions related to peroxide detoxification and biosynthetic reductive reactions, such as steroidogenesis. This is a summary of two processes scavenging peroxides, either organic or inorganic peroxides. And this removal of peroxides occurs at the expense of reduced NADPH. And the overreactions of these two systems are depicted in here. So glutathione and thioredox independent systems run at the expense of reduced NADPH. We have some enzymes that catalyze reactions that generate NADPH in cells. And it's important to remind that there is no described shuttle between the reduced state of NADP in the cytosol and the reduced state of NADP in the mitochondria, which means that what mitochondria need, it has to be generated in there. In the cytosol, the two major reactions generating NADPH are catalyzed by this enzyme and IDH1, whose substrates are glucose 6-phosphate and isocitrate. And this is the overreaction of NADP reduction. And in the mitochondria, we have 4 enzymes catalyzing reactions that generate NADPH. The two most important are those two in bold, IDH2, which oxidizes isocitrate, and the enzyme that's the focus of my presentation, NNT, which uses as a substrate NAG8. This is the NNT enzyme assembled in the inner mitochondrial membrane. The reaction catalyzed is facing the mitochondrial matrix. So this is an schema of the mitochondria with the two membranes and the matrix in here. This is the NNT enzyme, which catalyzes the transfer of a hydrate from NADH, reduced NADH, to NADP+, which is the oxidized form, thus producing the reduced NADPH. What's very special regarding this enzyme, and as we compare it to other enzymes generating NADPH, is that it's energy linked, because there is a channel in this protein that allows for the transport of protons down the gradient built by the respiratory chain, and this transport of proton displaces the equilibrium of the reaction towards the reduction of NADP. And this is very important because mitochondria keeps the redox state of NADP much higher than the redox state of NAD. And the experimental model we have been working with since 2010 is based on the well-known C57BL6J mice. And I would say that these mice, very famous, had an unfortunate career because they were established in Jackson Lab around the 50s. This is the first event in this timeline. And a second important event was the description of the full genome of this mouse strain in 2000. And from then on, this strain became much more used in many labs throughout the world because the genome was inscribed. But later, 5 years later, a group from England described that this mice strain are carrier of an NNT mutation. And at that time, nobody knew when this mutation had arisen. But a year later, it was estimated that the mutation took place in Jack's lab between 1975 and 1980. So it was about 25, 30 years of research, much research being done with a mutant mouse. As soon as we learned that this mouse strain had a mutation in this gene, we became interested in using them to study redox mitochondrial functions. And the first paper we published was in 2013. And I would say this was a very important event in this timeline because it changed a bit how people were using these mice. Because until then, nobody knew if this mutation had any consequence for mitochondrial redox functions. And in this first paper we published, considering the function of this enzyme, we compared the C57BL6J mice, which are mutant for this gene, to another mice strain that is wild type. But remind that these are two different strains. They might not differ only by the genotype of this NNT gene. So this is activity assay, which will show that the mitochondria from the wild type mice had the activity, but not the mutant mice. It's important to remind this was with liver mitochondria, and there were redox consequences. We evaluated some, but to summarize, I show you the redox state of glutathione, which was reduced in the absence of the functional NNT enzyme. The content was not changed, but the redox state was impaired towards a more oxidized one. Now I change a little bit. That data was from liver mitochondria. Now I present some more recent data we published in 2020 related to locomotor function in these mice at 2 different ages, when they are adult and when they are old. This is the results from a functional test in the rotarod, which was accelerating all the time, and the time to the first fall was measured, which is on the left, and the speed of the turning wheel at which they fell off the wheel is represented in here. We see here that when they are old, they are less tolerant to this functional test. So it indicated to us that there is some locomotor dysfunction. And we had 2 hypotheses. One is that the central nervous systems and neurotransmitters could be involved. We got some data on that, but I'm not going to show you. I would like to say that some neurotransmitters could be involved in this dysfunction, but we had to ask the question of whether any muscle dysfunction could play a role in this impaired locomotor function. This was not a straight question, because a group from Germany published in 2015 in Cell Metabolism some data showing that in skeletal muscle mitochondria, which is in here, there's no NNT protein, even the wild type mice. And they did not detect the activity as well. This is a positive control. Heart exhibits a lot of NNT activity. So we had to actually analyze if, in fact, skeletal muscle mitochondria have or not this enzyme. So we went on to use our model. I forgot to say that after the first paper, we backcrossed the mutated genotype into a wild type strain for 10 generations to generate a congenic control, which were used for these studies in vivo that we performed probably after 2015, when the model became available in our lab. Again, we assayed the NT activity. We could observe an NT activity in skeletal muscle mitochondria in this tracing here. In blue, we have the mutant mice. And on the right, we have a very important assay, which was performed with intact and respiring mitochondria, because on the left, we homogenized the tissue and mitochondria was permeabilized. And it's important to remind that this enzyme is energy linked. So having or not membrane potential is important for the function of this enzyme. So in here, we can see that the wild type mitochondria in black behave differently when a peroxide challenge is added to the system, and the mutant mitochondria didn't. I'm not going to go into the details of this protocol, but this is the fluorescence of NADPH. So we have a clear difference in here. For us, this was the proof that skeletal muscle mitochondria indeed exhibit NNT activity, contradicting that paper in Cell Metabolism. Not only mitochondria have NNT activity, but its absence changes peroxide metabolism. We have here some conditions of substrates challenging redox homeostasis in mitochondria. And we can see that in the absence of functional NNT, the rate of hydrogen peroxide release from mitochondria is very well increased under some conditions. This increased in hydrogen peroxide in the absence of NNT led to some signaling, probably because hydrogen peroxide has become a well-established signaling agent. And we see here that concurrent sources of NADP in mitochondria, which are the enzymes IDH2, which oxidizes isocitrate in the Krebs cycle, and malic enzymes. In the absence of NNT, they went up. These are young mice, adult young mice. Not only these NADP generating enzymes increased, but also superoxide dismutase SOD2, which is the mitochondrial isoform, also increased, suggesting that skeletal muscle cells were sensing this increase in hydrogen peroxide. And then we came to perform additional functional experiments with these mice at 3 different ages, young, middle age, and old. They performed a functional test, the hang wire test, and they had to cross a bar, hopefully without falling off. But as they are a little bit older, from the middle age, they started to fall more frequently from the bar. And at 9 seconds, these are the mean results in here. We have a lower performance in these tests, in the NNT mutant mice, and this is exacerbated as they get older in here. And the next step was to evaluate the bioenergetic function of this mitochondria. Of course, this is a mitochondrial enzyme that's both it's related to energy transduction, somehow related to energy transduction, and else redox balance. Here we have some data on the muscle mass of soleus, vastolateralis, and plantaris muscles. The decrease in muscle mass was only significant for soleus muscles at this age, almost 2 years old mice. Vastolateralis and plantaris didn't change the mass. And that finding is important to relate it to this other finding here, which is the this other finding here, which is the NNT expression in these 3 muscles, soleus, vastolateralis, and plantaris, all in the middle age. We see here that soleus muscle exhibits a lot of more of NNT compared to the other 2 muscles. Aging did not change the expression of this enzyme. This is the comparison across aging in soleus muscle, which was chosen because it had the highest expression. And these are said to us something, because soleus was the one that had the mass affected and is the one who has the highest expression among the 3 muscles. And in the respiratory analysis of this muscle, we found that oxidative phosphorylation was decreased from the middle age in the animals lacking functional NNT. Leaky or uncoupled respiration wasn't changed, and the maximum activity of electron transport chain followed the same pattern of oxidative phosphorylation, being reduced from the middle age. The vastolateralis had a similar decrease in the oxidative phosphorylation. The vastolateralis from the NNT mutant mice. but the plantaris did not show any difference in respiratory function between the genotypes. How much time do I have? 10? Okay. The next step was to evaluate muscle morphology, because we had that lower muscle mass. We had some problems with the preparation of slides to see the cross-sectional area. Actually, some tissue was cut longitudinally, and this compromised the evaluation of cross-sectional areas of a number of fibers that would be necessary to plot the histogram. But we could still analyze central nuclei in these muscle fibers, and it wasn't easy to do again the experiment, because we have to wait for 2 years for those mice to age. So central nuclei is a well-established marker of muscle degeneration. We can see here that in old mice we have a higher percentage of central nuclei compared to the control, wild-type control. And in young age, there's a trend, but it did not reach a statistical level in here. And this correlates to the functional test, in which the animal lacking transhydrogenase, the functional enzyme, seems to be weaker. And we did not put the data in this paper, the data related to muscle cross-sectional area, because we thought it was not reliable enough. But it was indicating muscle atrophy as well, which goes in parallel with the decreased muscle mass in solus. I would like to do some final remarks and some correlation with human conditions. In 2012, actually, we learned that there are some people, actually patients, carrying mutations in the NNT genes. At that time, we were about to submit our paper to Free Radical Biology and Medicine, and it helped a lot because it increased the interest people would have on this protein. And so far, it has been described more than 50 NNT variants that are pathogenic in humans if they are present in homozygosis or in compound heterozygosis. And the major condition caused by this mutation is a primary adrenal insufficiency. And there are also cases of impaired gonadal and thyroid functions in patients carrying these pathogenic variants. And some patients also exhibit cardiomyopathy, but it is more rare. Among the causes, the genetic causes of primary adrenal insufficiency, mutations in NNT probably explain 5% only. And most of the cases of primary adrenal insufficiency is caused by a defect in enzymes in the steroidogenesis. But what's the link between NNT and these endocrine disorders? It's pretty much related to the role of NADPH in steroidogenesis. Steroidogenesis includes some mitochondrial steps, some reactions that take place in mitochondria and require NADPH. So this is a possible link between mutation in this gene and the primary adrenal insufficiency, where in the cortex, of course, a lot of steroidogenesis has to go on. And oxidative stress caused by the absence of this enzyme would be an additional contributor to a degeneration of the cortex of the adrenal gland, which was shown in this paper in Nature Genetics in 2012. More recently, Roger Castilho, the PI in the lab at Unicamp, along with his PhD students, Annelise Francisco, worked with some patients and trying to characterize the functional consequences of mutations for the activity of the enzyme. And they published this paper last year. And for those who are more interested in human cases, I recommend reading this paper. I was not part of this one. And lastly, I would like to highlight that these pathogenic variants in humans were described in young patients. None of them were probably above 25, 30 years old. And there was scarce in-depth investigation of whether there are multi-organ dysfunctions in these patients. So we cannot say that, for example, skeletal muscle is not somehow affected, or heart, for example. And nothing is known regarding the effects of aging on the disease progress. I'm referring to the disease caused by this NNT variant, and our animal studies are the first to shed some light on this issue, because they indicate that non-affected organs during youth, which is the case, for example, of skeletal muscle, which seems to be the case for skeletal muscle, could become dysfunctional as the animal age. And this is it. I would like to acknowledge the contributions of Professor Aníbal Versesi, Roger Castilho. I have to say that Professor Aníbal studied this enzyme in the 80s, and this enzyme was discovered in the 50s, and it was a kind of lucky to be there in the lab when I realized in the literature that there was a mice strain lacking this enzyme, an enzyme that Professor Aníbal usually liked to talk to us about the function of it. Annelise Francisco, Claudia Navarro, Juliana Ronchi were 3 PhD students that carried most of the work presented in here. At the time of the earlier works, I was there as a postdoc in the lab. Now I'm in Ribeirão Preto, the School of Physical Education and Sport. And I would like to acknowledge FAPESP and CNPq for supporting these studies. And if you want to contact me, this is my email. I thank you your attention, and I will be happy to take some questions.
Hi, Thiago. Great talk. So I was curious about the heart phenotype of the B6NNT mutant line, because it's one of the major lines, mouse lines that's used for Chagas cardiomyopathy models. So if there's a mitochondrial impairment, it would be nice to know. And the second question is NNT expressed in immune cells? Because also B6 mice are known to have an enhanced inflammatory response with Th1 type profile.
Thank you. I'm going to answer the second one first. It is expressed in immune cells. Actually, the activity of NNT in that paper from Professor Roger, published last year, they evaluated NNT samples from peripheral mononuclear cells isolated from the patients carrying the mutation. And the first question is about the heart phenotype. And we haven't so far studied the heart deep enough. But that paper in Cell Metabolism from 2015, it was from a group that studied cardiac hypertrophy following the increasing afterload, a surgical procedure to increase the afterload. And they found that acutely, mice lacking NNT was protected against some changes induced by the increasing afterload acutely. But we suspect that chronically the final outcome, it could be a little bit different. But we suspect that they had performed some work on that, but they didn't publish it based on some results shown in conferences. And I think Professor Roger is right now planning on doing some research on the heart of these mice.
Thank you, Thiago. Very nice talk. Have you measured the lifespan of these mice? I mean, control versus NNT mutant?
No,
You
we
never
haven't.
did that. And how about high-fat feeding? Did you induce them?
Yeah,
Yeah. What do you?
we have done
Okay.
some challenges to them. If you put them on high-fat feeding, they not only have steatosis, but steatohepatitis, and the phenotype is much worse.
Okay.
Pyruvate dehydrogenase is only inhibited in the liver of these mutant mice when they are exposed to the high-fat diet. The control mice didn't exhibit this phenotype of pyruvate dehydrogenase inhibition, which further contributes to hamper peroxide metabolism, because in the absence of NNT, you have to have the first reactions of Krebs cycle running, because you need isocitrate to feed isocitrate dehydrogenase. So inhibiting pyruvate dehydrogenase, it's an additional issue in these mice lacking NNT. So we could rescue a little bit of this aggravated phenotype by using dichloroacetate. Someone said yesterday about the dichloroacetate. Who was? You. And we used it to relieve the inhibition of pyruvate dehydrogenase, and it worked.
Nice talk, Thiago. Thiago, I was told that these animals, they have insulin levels low. It's true? That's the first question, about the insulin secretion. And the second question is about is there any kind of compensation, I mean, for other antioxidant system?
Yes, thank you for the questions. I will answer the second first. There is compensation. I showed some increase in other mitochondrial enzymes that produces NADP8 in the mitochondrial matrix. That is in skeletal muscle. In liver, we didn't see any compensation regarding concurrent sources of NADP8, but in skeletal muscle, we did. And the other questions about insulin secretion, I think there are mixed results in the literature. You can find the result you want regarding that, because most of people that were trying to address this question of glucose tolerance in these NNT mutant mice compared different strains, mice strains. And this is a problem because there could be additional genetic modifiers not in addition to NNT. So we were the first to establish the congenic model. I think Helena Coutinho did some work on that. And if I'm not wrong, the insulin secretion is the same compared to the wild type, but they are glucose intolerant. This is, I think, well, better established in the literature. Thus, it's likely because a lower peripheral insulin sensitivity.
Another question. You said about human mutation, and some of them has been associated to cardiomyopathy. But do we have data on myopathies and something like mucosidosis or progeria?
No, you are referring to patients, actually. Yeah, no. There are only a few studies reporting cases of patients with carrying this mutation. All of them was discovered because some endocrine dysfunction. And as I said, they did not investigate in depth if other organs were affected or not. At least it wasn't reported in those studies.
Because I had the feeling when you are looking for progeria, it's a high-speed aging disease. So the children, after 4 or 5 years, they look like 60 years old. So maybe some NNT variant may be involved also.
Oh, cool. Thanks for the suggestion. I'm going to look at that.
Thank you, Thiago. What were the implications of the identification of the mutation for the literature? Because the model was established in the 70s, right? And the mutation was identified like 30 years later. And how the literature reacted to the identification, and what was the Jack's response?
Jackson is messing up with a lot of models, because if you go there to buy a transgenic or a knockout, they usually recommend which is the control mice. And for a lot of these genetic modified models, they recommend the C57, 57BL6J mice. But a group from Canada ordered probably almost 100 mouse from different strains from Jackson, and they genotype them. In most cases, the NNT genotype of the control strain recommended by Jackson is different from a transgenic model or a knockout model. The community tried to not to respond at first, I think, to this description. The first time I went to a conference, I think it was experimental biology with this data in 2009, I think, or 2010. People in US were running away from the poster because a lot of them have an animal model in the lab that is probably a carrier of this mutation. So if you change, for example, the response to high-fat diet, how could we use this model to study high-fat diet, assuming it is a model of a human condition, considering humans have transhydrogenase?
So we will arrive to the end of this session. So I can call our 4 speakers, Luis, Thiago, and Fernanda. So you can come. That's your last chance to ask question or discussion with the speakers.
So, Professor Thiago, did you take a look? Hello? Okay. I try to do this. Professor Thiago, did you take a look in the NAD levels on these animals? Because in your enzyme, they're actually regenerating NAD, and then when you have a knockout on this enzyme, probably they have a reduced levels of NAD in the whole system, mitochondria in the organ or in the part of the tissue that you are evaluating.
Yes, in the first paper in 2013, the content of NAD, both in the reduced and oxidized forms, and the content of NADP were measured, but was in liver mitochondria. In that sample, in those samples from liver, there was no difference for the content of these two coenzymes.
Correct. So did you take a look in the also in the glycolysis system? Because if you have like a regeneration of NAD, they can boost glycolysis, and you saw a reduction in oxphos in older animals. So did you take a look in glycolysis in these older animals?
No. We haven't been able to study much the older animals because it's hard to have mice at that age. So we did what was possible with the few animals we got at 22 months of age.
If there is no additional question, we can close this morning session. And I hope to see you this afternoon for the last session of this workshop. Thank you to the speakers.
Som.
Você tem chave aqui? Não. Se bater. Se bater, ele fecha automático. Ah, não, mas você conseguiria depois sair, entrar? Sim. Ah, a chave não tá comigo. Por isso que eu tô falando. Cara, você vai na porta, então vai depois. Beleza, então, combinado. Valeu, obrigado, hein?
Som, teste. Testando. Oi, som, teste. Um, dois, teste de áudio. Teste. Som, teste. Testando. Som. Oi, som, teste. Um, dois. Testando. Som, teste. Um, dois. Teste. Testando. Som. Teste de áudio.
Good afternoon. Good afternoon. My name is Deborah Levy. I will be the chair now at this session. I would like to present Dr. Elena Saranova. Dr. Elena is a scientist by training and an entrepreneur at heart. Currently, the founder of NMN Bio, she's redefining healthy aging for people over 30 with a longevity supplement range that offset physical and cognitive decline. Be welcome.
Thank you so much, everyone. 1-1. Can you hear me well? Okay, amazing. How are you guys doing today? Okay, so I'd like to start with asking you a couple of questions, if that's okay. So I would like to know how many of you are undergrad students today. So I just have a feeling for the audience. Just raise your hand if you're an undergrad student. Amazing. And how many of you are postgraduate students? Awesome. Okay. So I will tell you my own personal story, how I switched from biology studies to funding my own company. So back in the days, I did my PhD with Dr. Sovan that gave a lovely talk yesterday, and I was studying NAD metabolism and how it's related to neurodegeneration, how it's related to autophagy, how do these two intersect, basically autophagy and NAD metabolism, and how this is applicable to human disease and human health. So basically health span and lifespan. Basically, after we did an unbiased metabolomics approach to see what is affected in the autophagy deficient neurodegenerative phenotype that we were studying, we saw that NAD and NADH were depleted in our stem cell derived neurons. So we kind of started looking into NAD metabolism and what are the implications for those cells and how we can restore it. So interestingly, what we found is that even when autophagy is deficient, which is something that we're seeing in all neurodegenerative disorders, if you do boost the levels of NAD in the cells, you can actually rescue the phenotype. So that was very interesting. And back in the days when I was actually, like a teenage girl and I was dreaming of becoming a supermodel and having a cover in Vogue magazine, I didn't know that actually my cover girl moment would come at the Journal of Molecular Biology with the stem cell derived neurons that I differentiated. And we got this beautiful cover back in 2023, I believe. So what was happening during that time in the lab is that I was obviously rushing to finish up my PhD thesis back in 2019. I was working a lot of hours in the lab, so 16, 18 hours a day. So I was tired, and then I realized that, wait a minute, I'm already treating my cells with some NAD boosting compounds that actually have amazing effects in the cells. And I realized that there are companies out there that are already selling those NAD boosters as food supplements, because a couple of them, such as, for example, NMN, nicotinamide mononucleotide, are not patented. So it's a generic compound, just like vitamin C, let's say. So someone can basically sell it. And this is what they started doing back in 2018, 2019. So I basically went to Amazon UK and ordered some NMN for myself. And I was fascinated by the fact that 2 days later, I wasn't tired anymore. So this was absolutely mind-blowing for me because I've never experienced such a profound effect from a supplement before in my life. And then after a month, when the bottle has finished, I wanted to buy some more for myself. So I went back to Amazon online, Amazon UK, and then this vendor was out of stock. So I bought the NMN from another company. And then another month is passing by. I'm feeling fantastic. I'm generating all this beautiful data. And then what happened is that I go back to Amazon after another month, and now this vendor is out of stock. So I realized that actually there is a lot of demand for this kind of supplements, but there is not enough supply. And this is when different kinds of publications about NMN in particular started coming out. They found the SLC 12A8 transporter, which basically allows the NMN to enter the cell, and it converts into NAD into one simple step. Because before they discovered the transporter, the Medical School of Washington, they didn't know what is the mechanism of action, which NAD precursors would be the most efficient one into boosting NAD, but actually they realized that, for example, if you take another one, NR, nicotinamide riboside, you would need it to get converted into NMN first, and then the NMN would boost the NAD. So the science started coming all together, and there are a couple of human clinical trials of efficacy and safety. They started coming out during that time, and I realized that, okay, well, it looks like the compound is gaining a lot in terms of popularity, and there is a lot of demand for it. And also, so I found a, let's say, product market fit. But also what I realized is that I found a founder market fit, because I was literally doing my PhD in NAD metabolism. And this is where the puzzle came together, and I realized, you know what, I actually can start a company selling a high quality product, because the other problem back in the days was that there were a lot of companies that would just white label the NMN and it wasn't good quality. You would message them to get the certificates of analysis, no one would reply. So then I saw the gap in the market, basically, and I founded the company back in 2020. So this was during the pandemic, and I had to stop my my PhD work in the lab back then, because you know, we have to shut down the lab basically. So I was just sitting in my apartment by myself, wrapping up my thesis. Fortunately, I had enough data to finish my PhD and defend my PhD thesis. And at the same time, I started doing market research about supply chain and production manufacturing. How can I even source this compound? What would it take to manufacture it, to produce it at a high quality in a GMP certified good manufacturing practice in the UK? So I was putting together all this stuff like for a few months. And then in December 2020, I defended my thesis, and I think that a couple of days later, the website went live, literally. I think it was literally the next day, something like that. So I started the company with just 2 SKUs, so 2 products, basically, just the 2 NMN products, and then we expanded from there. So we started as an e-commerce company, but we expanded quickly into selling globally, online, basically. So we have a warehouse in the US now. We have another one in the UK, in the UK, of course, and then in Europe as well. And we'll be setting up another one in the Middle East soon. And then what happened in 2023 is that Aster Pharmacy approached us. So Aster Pharmacy is the biggest pharmaceutical conglomerate in the Gulf countries, in the Middle East. So they have over 250 pharmacies in the UAE. And back in the days, because the compound was trending, they were actually looking for a reliable supplier of NMN from Europe or UK. So they were screening the various companies that were selling NMN, and they actually chose us. So we didn't even approach them. They approached us because of our reputation and our evidence-backed supplement range, and the fact that we have a completely transparent supply chain. So for example, if someone messages us, we will disclose all of our suppliers, all of our production, manufacturing processes, and so on. So this is written on our website. So they approached us, and of course my scientific background helped, I believe, in their choice. So we launched with them back in 2023, which actually had a kind of a compounding effect for other retailers worldwide as well. So we would message another international pharmacy chain from another country saying, look, we're already in Aster Pharmacy in the UAE. So this kind of created the trust and the confidence for other suppliers to take us on board as well. So UK is still our base. So we still are producing all of our supplements in the UK, because we don't compete on price, we compete on quality. So we could potentially produce for cheaper at another country, but we don't do that because actually all of our international retailers love the fact that we are producing our supplements in the UK. So now this is snowballing. Of course, we have some esteemed retailers in the UK as well. So Selfridges, for example, is stocking us. We're launching with Boots UK later this month. The same time, we are developing our down to consumer path as well. Like we launched the mobile app where you can download it, by the way. So if you just type in NMN Bio into the App Store and Google Store, you will find the app. We have a brain game to evaluate your cognitive function and so on. And now what happens is that our wholesale arm of the business has overtook the down-to-consumer path. And in November 2024, we got featured in Financial Times. So usually this kind of press release is costing a lot of money, and you have to have a connection, and they have to, you have to go through a timely process. But we actually got this for free because Financial Times wrote an article about longevity and NAD supplements and like the different form of NAD boosting activities, basically. I say it like that because beyond oral supplementation, you can do IV drip, for example, with NAD, but this doesn't do much, which is what Financial Times wrote about. And out of the two companies they chose to feature worldwide, one of them was NMN Bio. So this gave us an additional boost and opened the doors for a couple more retailers. Do have a milestone there, launching in 6 countries, so this has already happened, and we're launching with Saudi Arabia later this summer as well. So all these features basically add the credibility and the confidence to the company, and our bestseller product is still the NMN, and this boosts our growth in terms of launching additional products as well. So now we have 5 separate products, 6 SKUs, because we have 2 quantities for the NMN, and they all target different hallmarks of aging. And actually we are collaborating with various labs worldwide to generate scientific data on the different indications. So we're still collaborating closely with Dr. Sovan in Birmingham. We are looking into organizing a human clinical trial as well. And if something from the slide caught your attention with regards to what you are working on, please come and talk to me. We would be open to providing some compounds for collaborations, including in Brazil and in other countries as well. So yeah, so this is our partners, and at this point I'm usually getting the question of whether it's worth basically pursuing further scientific studies, postgraduate studies, and so on, to gain more scientific expertise if someone wants to be an entrepreneur. And actually the priceless thing for me from my PhD, beyond the scientific knowledge that I gained, is that postgraduate studies basically teach you how to think. And this is something that is a fully transferable skill to other areas of your life, including entrepreneurship. And actually, I think that scientists are uniquely positioned to start businesses because they're already used to be operating according to rigorous protocols, for example, right? So because if you don't do that in the lab, everything will fail, as you know. So I think that scientists are actually having like naturally curious minds, and they develop resilience and other skills that will that is a great foundation basically to start their own venture. So why you should consider entrepreneurship is another question that I get asked a lot. And basically, I think that during your scientific training, you would be able to discover, you would be able to notice different things in your own workflows and in the experiments that you're doing, and with everything that you can come in contact with in the lab. You would be able to discover different bottlenecks and different things that could be improved commercially, right? So for example, I'll give you another example. So back in the lab, I was actually looking into how to optimize freezing reagents for a specific kind of cell type. So I came up with this idea in the lab, and then I optimized my own freezing medium. I didn't end up commercializing that idea, but this is just an example. And I'm sure that each one of you comes up with different ideas of what can be improved in the lab on like a daily or a weekly basis. Like I used to have a lot of those ideas when I was doing my PhD. So you basically combine your passion, which is probably already, you know, like the field of biomedicine, and then what are you good at and your specialized knowledge, and then you also apply a third filter, which is how would it drive your economic engine, so how it can actually produce profit. So those would be the questions to ask yourself. And then you would tell me, okay, Elena, so maybe I have an idea and I want to start somewhere. Where do I start? And actually, it's much, much easier than you think, because you already have developed the skills of, let's say, detailed project management, right? So the first step would be just to Google it or put it in ChatGPT, and it's actually admirable what you can find online nowadays. So this is what we were talking about yesterday as well, that the barrier to entry into any venture right now is as low as it can get if you have access to the internet, because you can literally ask ChatGPT, okay, I want to find a supplier of, I don't know, soft gels in South America for a supplement company, and ChatGPT will literally give you the emails and the contact details of the top companies that do this, that create this particular product, right? So it's actually easier than you think, because again, you've already been trained to be finding and analyzing scientific information, detailed information during your research work, right? And then, so yeah, so there could be different kind of things involved in various projects, obviously. Another one we also talked about with someone yesterday is that you have to surround yourself with a stimulatory environment that will be motivating you to basically innovate. But here the pleasant surprise is that, again, if we're having access to the internet these days, we can literally follow a few entrepreneurs on Instagram and be consuming their content for a few minutes a day, and therefore integrate, you know, the this kind of entrepreneurial mindset into, like, give yourself a daily dosage of this entrepreneurial mindset online. So there's literally no excuses, I think, nowadays, because we have access to so much information. And of course, the other one that unfortunately they don't teach us at the university is how to keep yourself mentally and physically fit. This is very, very important as well. But again, we have access to so much information nowadays that there is no excuses. So, transferable skills from biomedical research to entrepreneurship. You already have the patience and the discipline and the humility, and your brain is highly analytical, right? So you know how to create protocols, you know how to split test things, you know how to create SOPs, standard operating procedures, and how to do the problem solving. So you're already there. And then it's the same kind of skills that you need for your to start a business of your own, basically, right? And then, of course, the optimal health. I'll keep on stressing this out, because without optimal health, you won't have maximum results in your career and other areas of your life. So this is what happens when you implement tiny improvements every day. So even if you allocate just 5 or 10 minutes a day to your fitness, or to do your scientific reading, or in parallel with your research to start also searching up how you can set up a company, right? So you can develop those skills just by integrating 5 or 10 and 20 minutes a day in a specific hour to this particular project, right? And this is what happens, like, you can improve everything this way, right? And the result of the compounding interest could be positive, or it could be negative, you know, so some bad choices will stock up as well, and then you're going to have a negative result. So, and the other thing is that you have to be asking yourself the right questions. So what I hear a lot is people telling me, oh yes, but I don't know how to do this. Oh, but this is difficult to do. So for me, I always ask myself, okay, how can I do this, right? So I already assume that it's been like I can do it, and I just need to find a way to achieve a potential target, whether it's finding some financing for your company, or a scientific problem, or something else. And then the other question that I find very, very useful is, has anyone else done it before? Because again, we had a conversation yesterday and someone told me, oh yes, but like I'm by my own, like how can I do this? And I said, okay, well, has anyone else founded a company by themselves? If the answer is yes, then this means that it is possible, right? So you just need to find a way to do it. Okay, creating the environment, we already talked about it, and then you're integrating on a daily basis, because it doesn't matter what kind of brilliant ideas you have, you actually need to make sure that you're basically defining a trajectory for yourself. So let's say you want to say, I want to open a company in a year from now, right? Like I have a brilliant, amazing idea, and in a year from now, I want to be a business owner and I want to be selling this product or commercialize it somehow. Okay, well, you you break it down into monthly targets and you say, okay, well, so what does this, like, what do I need to do in 6 months from now? What do I need to do in 1 month from now? And then you bring it down into tiny little decisions that you take every day. So the person that opens up a company in 1 year from now, what do they do today, right? Do they go, I don't know, like out drinking every day? Like probably not, right? So you have to kind of align your reality and your daily actions with what you want to achieve in the future. And you can go as micro or as macros as you'd like. And then, of course, to think what other variables I have control of, because many times we think that our first initial reaction is that, yes, but I don't know how to, again, like an example, how to get financing, right? But like, how much time have you actually invested into searching this up, into evaluating different sources of financing across, you know, various, across the your preferred subject of interest. Let's just put it this way. So you already have built an amazing character as a researcher by doing biomedical research. And like, because I went through it myself, like I know how hard it is, right? And actually, like at this point, I have like, I have good news for you, because biomedical research will be the hardest thing you'll ever do in your life, right? And then after that, it will, everything else will be easy. So like, give me any company to run right now. Like, it's so easy. You just put the pieces of the puzzle together and you create protocols and you follow the protocols, right? And again, like regular people that are not researchers, they don't think like this, they just do random stuff, right? So we already have a huge competitive advantage as researchers in what we're doing, right? Because compare myself to someone, to another, let's say, I don't know, food supplement company founder that doesn't have the PhD, right? Like I already have such a huge competitive advantage in terms of how I think, how I structure my workflows every day, and how I manage the project, right? Because I already have this experience from my research. So, and then of course, a lot of entrepreneurs want short-term results and they get demotivated very fast, but that's not how we act. That's not how researchers act, because we are learning to operate to operate for a long time without short-term reward. Like I know that it was like this for me during my PhD. I was working with stem cells, so we were creating, you know, like we had some cell lines that I was differentiating into neuronal cells for the first time. So we had the human embryonic stem cells that Sovan talked about yesterday with the genome engineering, with the ATG5 mutation, so they don't have autophagy. So actually they're not very, you know, they're not very happy cells, and I had to differentiate them into neuroprecursors and neurons, and of course it would take weeks. And then imagine, you know, something goes wrong and you mess up the experiment after like 3 or 4 weeks of cell culture, and then you have to start all over again. And again, like this is my personal example, but I'm sure you have other examples of very lengthy workflows that you have to deal with in your research. So regular entrepreneurs have never done something like this before, and this is mind-blowing because you already know how to operate this way. So a very profound competitive advantage here for the scientists. And then, so yeah, this is what I said, like the bar to success is actually very, very low, because when you're looking at commercial ventures where the founders don't have the research background, like they will be operating in a completely different way. Something that we are also learning is like we learn how to work on long-term periods of time by ourselves, which is amazing. And then this is helping with the entrepreneur's paradox, right? So we are eagerly optimistic about the future while not needing gratification in the present. And this is an amazing mindset to have. And then, of course, another thing that comes into play is like how you prioritize basically your time. What do you prioritize every day and every week? And then you build the foundation, the thorough foundation for your business this way. And as I said, you know, like we actually can modify our reality much more than we think, and we can basically influence the output by the input that we're putting into a project. Okay, all right, stimuli, and yes, so we already know how to enjoy the process, and this is absolutely great for being a founder, basically. So I have some book recommendations. Please feel free to take a picture of this slide. And I actually have a mix of habit-forming books, business books, and actually health and wellness books. And one book that basically changed my life back in 2019. So true story, I was feeding the neurons in the lab with my headphones on, and I was listening to the Why We Get Sick book. So this book was actually changed my view on nutrition and on, like how I basically treat my body, because every chapter basically has a name of a different disease. So let's say cancer, neurodegeneration, diabetes, and so on. And then actually each chapter explains to you how this is linked to insulin resistance. And once you see it, you can't unsee it. So I always recommend this book to everyone because you can understand your own biology much better. And then if you hack your own biology, you can actually, like with your nutrition and other lifestyle interventions, you can hack your productivity levels. You can hack your cognitive function and your productivity, your physical performance, everything, right? And then you can be a better professional. So it's all connected, like it's all together. Deep Nutrition, another amazing book on actually the evolution of the human species and how the human body was changing depending on the nutrient availability. So, and then a couple of other books that have great business advice as well. Okay, so as an entrepreneur, this is me flying over Rio a few days ago. There are a lot of freedoms to conquer in this life. One is location freedom. So this one I conquered a long time ago. Like I can run my business from anywhere in the world. So usually I split my time between Dubai and London, but like I can allow myself to travel. Sometimes I go for a couple of weeks in a jungle in Thailand, and I just sit there and meditate and work on my business. Financial freedom is something that I'm conquering at the moment, and then time freedom. This is going to be the most difficult one to conquer, because as the business scale, it actually keeps on requiring your attention. But it is what it is. And you know what? For me, I don't feel like I work a day in my life, because like every day I wake up and I'm excited about growing my business and making an impact into our customers' life by improving their health. So the moral of the story is that actually you can't fail if you never quit. I really love this saying, because if you just keep on going, you keep on increasing your chances of success. And we also have another saying in Russian, which is the death catch. So for example, when you have a dog, like a pit bull or something, and they catch a prey, so they don't unclench their teeth until the prey is dead or the dog is dead. So this is the death catch. And I think that when you look at your goals this way, that I'm going to get it done no matter what, you significantly are increasing your chances of success. So as I said, we are looking for worldwide collaborators. Like, we are very happy to collaborate with academic labs. If you think that some of our compounds could be useful for your research, we can discuss. Please come find me later if you'd like, or please reach out for collaboration. And thank you very much for your attention. Everyone is shy. Okay.
Thank you very much.
Good afternoon, everybody. Thank you for the kind introduction. I also would like to thank Adesio and everyone in the organizing committee for the invitation to be part of this great workshop. And today, I'm going to talk about the role of metabolite as signaling molecule. But before I start, I have to say that the concept that metabolites can function as signaling molecules is not new at all. It's already well described that metabolites can regulate their metabolic pathways by direct binding to proteins. For instance, the enzyme pyruvate dehydrogenase that converts pyruvate into acetyl-CoA is inhibited by pyruvate dehydrogenase kinase, and those kinases can be inhibited by direct binding of pyruvate or activated by acetyl-CoA. And by doing so, those metabolites can regulate their metabolic pathway. So if it's not new that metabolites can function as signaling molecule, what am I doing here today? Well, in the past years, some research groups have shown that metabolites can do much more than that. They can also regulate metabolic biological process outside metabolism. And to give you some examples about it, today I'm going to talk about two recent discoveries that we have made in our lab. So in the first part of my talk, I'm going to show you how lactate controls cell proliferation. And in the second part, I'm going to show you some data that suggests that succinate regulates muscle remodeling and muscle adaptation to exercise training. So it was described many years ago that a high rate of lactate production from glucose, despite the availability of oxygen for mitochondrial respiration, is a hallmark of rapid cellular proliferation. And this metabolic phenomenon, known as aerobic glycolysis or Warburg effect, has long been associated with the proliferation of cancer cells. But what is interesting about this is that lactate was for many years considered a metabolic end product of glycolysis. But for us, it doesn't make too much sense, because why would a proliferating cell waste all the carbons in the lactate molecule? So here we considered whether lactate could play a role as a signaling molecule and somehow regulate the cellular proliferative state. And to test this hypothesis, we used a thermal proteomic profiling approach. So in this experiment, we used hexyl lysates treated with vehicle or 2 concentrations of lactate. We heated the cell lysates at different temperatures. There's a problem here. And then we performed mass spectrometry analysis to determine protein term stability. So the rationale behind this experiment is that if lactate binds to a protein, it will increase its thermostability. And we verified that lactate treatment increases the thermostability of several proteins. Most important, UB2C was among the proteins exhibiting the most robust increase in thermostability in response to lactate treatment. For those who don't know, UB2C is a ubiquitin conjugating enzyme that interacts with this protein complex here, the anaphase promoting complex. So once the polyubiquitin chain is conjugated by UB2C, it's transferred to the anaphase promoting complex that later transfers it to proteins that regulates mitosis. So the anaphase promoting complex targets protein to be degraded by the proteasome. And by doing so, it regulates cell cycle progression and promotes cell proliferation. However, it's important to mention that a non-covalent binding between small molecules and proteins usually does not cause a large shift in protein thermostability like this one observed for UB2C in response to lactate treatment. So it's quite likely that these finds results from a protein-protein interaction. In other words, it seems that the increase in UB2C protein thermostability is not caused by a direct bind of lactate to this protein. So here we hypothesize that lactate could somehow facilitate the interaction between UB2C with its partnering DNA promoting complex. And to test whether that was the case, we treated cell lysates with lactate or the control condition. We immunoprecipitate the anaphasine promoting complex and blot against UB2C. And as you can see here, lactate treatment increases UB2C binding to the anaphasine promoting complex. But how does lactate do it? In order to answer that question, we again treated cell lysates with lactate, immunoprecipitated the anaphasine promoting complex, but now we performed proteomics analysis. And we verified that lactate promotes a massive increase in the summoylation of the anaphasine promoting complex. Indeed, it was already known that the anaphasine promoting complex subunit, APC4, can be summoylated at 2 sites. And this post-translational modification promotes the activation of this protein complex, which contributes to cell proliferation. However, protein summoylation is a reversible process, like phosphorylation. So the sumo present on APC4 can be removed by the desummoylase enzyme SEMP1. So here we thought that SEMP1 could be a target of lactate, and by inhibiting SEMP1, lactate would increase the summoylation of the anaphasine promoting complex. We did a lot of structural biology experiment, and I'm going to make a long story short. So we found that lactate binds to SEMP1 active site, and if we mutate lactate binding site, this metabolite is no longer able to inhibit SEMP1 activity as you can observe here in wild type SEMP1. Most important, SEMP1 inhibition is required for lactate-mediated anaphasine promoting complex summoylation. For instance, if we mutate lactate binding site on SEMP1, this metabolite is no longer able to promote the anaphasine promoting complex summoylation, as you can see in these representative plots. And to test whether lactate-mediated the anaphasine promoting complex summoylation is important for UB2C binding to the anaphasine promoting complex, we mutate the summoylation sites on APC4, and then we treated cell lysates with lactate, immunoprecipitate the anaphasine promoting complex, and blot against UB2C. And we observed that the effect of lactate on promoting the interaction between UB2C and the anaphasine promoting complex is lost by mutating the summoylation sites on APC4. So overall, these results suggest a model whereby lactate-mediated summoylation of the anaphasine promoting complex leads to a structural reorganization of the anaphase promoting complex that is permissive for UBE2C binding to it. Finally, we verified that lactate signaling it's important for cancer cell proliferation. For instance, if we the reduction of intracellular levels of lactate by treating cells with dichloroacetate or by expressing the bacterial enzyme lactate monooxygenase impairs the proliferation of two cancer cell lines. And similarly, if we disrupt lactate-mediated simulation of the anaphase promoting complex by mutating the simulation sites on APC4, cancer cell proliferation is also impaired. And we tested both conditions in the context of hypoxia to mimic the tumor microenvironment, and basically we saw the same result. Disruption of lactate signaling impairs cancer cell proliferation. So the take-home message of the first part of my talk is that elevated glucose catabolism drives lactate production, which inhibits SAMP1. By inhibit SAMP1, lactate increases the anaphase promoting complex simulation, which facilitates UBE2C binding to it. And by regulating all those processes, lactate contributes to the cellular proliferative state and the proliferation of cancer cells. Of course, we still have several open questions about lactate signaling. Now we are focused on understanding whether lactate regulate the simulation of other proteins in addition to the anaphase promoting complex. And here we are particularly interested in the heart for several reasons. First, cardiomyocytes are post-mitotic cells that usually consumes lactate instead of producing it. However, Edesio showed us very nice data yesterday that demonstrate that a hallmark of failing hearts is a metabolic shift towards glycolysis. So cardiomyocytes produces lactate under pathological condition. These are very preliminary data, but to test whether lactate regulates the simulation of cardiac proteome, we treated heart lysates with lactate, immunoprecipitate the simulated proteins, and then we performed proteomics analysis. And we did this for SUMO 1 and SUMO 2, 3, and the result was quite similarly, actually. And we verified that lactate indeed increases the simulation of some cardiac proteins. And here I highlighted triadine and juntofilin 2 because they are because those are proteins very important for the calcium handling process. And there are evidence in the literature that malfunction of both proteins cause cardiac dysfunction. So now we are trying to understand what is the impact of lactate-mediated simulation on the activity of both proteins and the role of lactate signaling in the pathophysiology of heart failure. So now I'm going to switch gears a little bit to talk about how a mitochondrial metabolite regulates skeletal muscle remodeling and skeletal muscle adaptation to exercise training. So when you think about muscle adaptation to exercise training, it's a very complex biological process that involves not only hypertrophy of muscle fibers, but also neurogenesis, angiogenesis, extracellular matrix remodeling, and so on. And usually those processes are modulated by paracrine factors released by muscle fibers during exercise. However, the signals that controls muscle adaptation to exercise training are still poorly understood. And because muscle remodeling begins rapidly upon acute exercise, we hypothesize that metabolites originating from exercising muscle could promote muscle adaptation to exercise training. And to test that hypothesis, we performed metabolomics analysis in the whole muscle, in the muscle extracellular fluid, and in the bulk plasma of acute exercise at mice until exhaustion. And to be considered a muscle-released metabolite, as such metabolites should fulfill some criteria. First, be increased in the extracellular fluid post-exercise, be increased in the extracellular fluid post-exercise when compared to whole muscle, and be increased in the extracellular fluid post-exercise when compared to bulk plasma. And only a single metabolite fulfills all three criteria, which is succinate. And this result is quite unexpected because the plasma membrane of most mammalian cells is thought to be impermeable to mitochondrial decarboxylates, including succinate. And as you can appreciate here, we verified that that the secretion of lactate by muscle fibers during exercise is also conserved in humans. For instance, lactate levels in the femoral artery and vein increase more than tenfold during exercise, but it returns to baseline levels few minutes after the end of the exercise section. But how does lactate get out of muscle fibers? What is the mechanism behind lactate secretion by muscle fibers? Succinate, sorry. It's too many metabolites in my head. Sorry. Yeah, succinate. So we verified that exercise increases glycolytic flux, which causes a reduction in the cytosolic pH. And because of the relatively low pKa of succinate, this condition renders a substantial proportion of succinate in the monocarboxylate form. And in the monocarboxylate form, succinate can then be transported out of muscle fibers by the monocarboxylate transporter 1. Succinate is known to be a ligand of a G-protein coupled receptor known as succinate receptor 1. So here we considered whether muscle-released succinate could regulate muscle adaptation to exercise training through its cognate receptor. Even though we can detect succinate receptor 1 expression in whole muscle samples, muscle fibers do not express succinate receptor 1. For instance, we can clearly see in this muscle in silico hybridization experiment that succinate receptor 1 transcripts, labeled here in red, colocalizes with non-myofibrillar muscle cells, labeled here in blue, but not with muscle fibers, labeled here in yellow or green. And to verify whether succinate released by muscle fibers during exercise initiate a response on non-myofibrillar muscle cells, we isolated the major populations of non-myofibrillar muscle cells from acute exercise wild type and succinate receptor 1 knockout mice, and then we performed RNA sequencing analysis in those cell populations. And we verified that several pathways known to be important for muscle remodeling and muscle adaptation to exercise training, including regulation of neuronal projection, axon guidance, TGF-beta signaling, and DGFR signaling, is down-regulated due to knockout of succinate receptor 1. And to test whether succinate signaling in non-myofibrillar muscle cells is important for skeletal muscle adaptation to exercise training, we subjected wild type and succinate receptor 1 to exercise training. It's not actually exercise training, it's wheel running, but it's a model of exercise. And we verified that succinate receptor 1 knockout mice exhibit a striking absence of muscle strength gains, unlike the substantial gain observed in wild-type animals. And similarly, exercise induces a massive increase in muscle innervation that is abrogated by knockout of succinate receptor 1. So, those finds suggest that muscle-released succinate is important for muscle adaptation to exercise training by promoting muscle innervation and muscle strength gains. And now we want to understand whether other tissue respond to succinate released by skeletal muscle during exercise. For instance, we performed proteomics analysis in the heart of wild-type and succinate receptor 1 knockout mice subjected to acute exercise. And we saw that several pathways known to be important for cardiac function, including regulation of heart contractility, response to calcium, sarcomere organization, regulate of heart rate, and others, were also downregulated due to a knockout of succinate receptor 1. Even though these are very preliminary data, it suggests that muscle-released succinate may also be important for cardiac adaptation to exercise. And I hope to be able to bring you more data about this story in the next opportunity. With that, I would like to thank everyone involved in this project, especially Edward Ciociani, who was my postdoc mentor at Harvard Medical School. Most of the work that I have shown you here today was done in his lab. Our financial support, my research team, and thank you very much for your attention. And please, if you are interested in metabolite signaling, or if you know somebody that might be interested in this topic, please contact me. We are looking for all kinds of students to join our lab. Yeah, thank you very much for your attention, and I'm happy to take questions.
Thank you very much.
Sure.
Questions?
Beautiful work, boss. Congratulations.
Thank you.
Very interesting. There's some papers in the literature showing recently that lactate and even succinate then can be attached to proteins and to work as a post-translational modification. So how do you see that fitting in the work that you are doing?
Yeah. Indeed, the succinate and lactate can bind to protein and promote post-translational modification. Yeah, I mean, it's another way that those metabolites can regulate like biological function. Here, like especially for succinate, we were interested in this intertissue crosstalk through the succinate receptor 1. But it can also be uptaken by cells, promote not only post-translational modification in proteins, but also in the case of succinate, induce like ROS production and so on. So there are many ways that metabolites can regulate biological function. It's very hard to study metabolite signaling, especially if you don't know, especially if it doesn't have a receptor.
Sure.
Very nice talk. Congratulations.
Thank you.
So I wonder, is there a time-sensitive effect of exercise and production of and release of succinate? Because I would expect that the liver would uptake it after some time or still during the actual bout of exercise. Have you looked at this time-sensitive aspect?
Do you mean like the time that lactate remains upregulated?
Succinate, yes.
Succinate, sorry.
Yes.
Oh. In the circulation? Yeah,
Yes.
at least in humans, it's, yeah, I guess I show you data. It's around like 30 minutes, but it's not uptaken by liver. Ed Shouchani showed that like when you inject succinate in the blood, the main tissue that uptakes succinate is brown adipose tissue.
But even in humans?
Oh, not humans. He did that in animal models, in mice, but I'm not sure about humans. I'm not sure if like anyone have done that. Yeah. But of course, we don't know how extracellular concentration of succinate is regulated, but now we know that one tissue that is involved in that is brown adipose tissue. But like, for example, other tissues can uptake that.
Okay. Thank you very
sure.
much.
Very, very nice. Congratulations. And my curiosity is about the possible inflammatory effects of this persistent release of succinate from muscle. Might it be related with like overtraining symptoms or the muscle damage, this kind of thing? Do you have any evidence of that?
Yeah, that's a great question. I mean, yes, succinate is pro-inflammatory, and it seems to be good when you have this transient elevation. So, like, you go for a walk, for a running, and you increase the succinate levels in the extracellular media, and then we stop, it goes down. But like under pathological condition, like for example obesity, it's sustained elevated. So in this scenario, it's deleterious, and it's pro-inflammatory. So indeed, it can be deleterious. And in our lab, we are also trying to understand, for example, in the context of obesity, if sustained elevation of succinate impairs, for example, heart function, you know, because during in the pathophysiology of heart disease, we have like, for example, a reduction in the brown adipose tissue, and this can like compromise the capacity of our body to uptake succinate from extracellular media. So indeed, if you like keep the extracellular concentration of succinate up, it's deleterious. Regarding overtraining, it's hard to say because the secretion of succinate is not like related to muscle damage. For instance, in our experiment, we didn't see like this. How can I say? Disruption of muscle fibers and anything like that. So it's selectively released. But when you have overtraining, you're going to have like muscle damage, and you have like a lot of molecules that just go out passively from muscle fibers. So it's hard to say if succinate or any other molecules, like for example, mitochondrial DNA and other molecules, they will be able to trigger like an inflammatory response and contributes to the overtraining effect. Of course, succinate can play a role in that, but again, it can be uptaken by other tissues. So I guess that even if you like have an overtraining condition, after a couple of minutes, it will be uptake, unless you have like a pathological condition.
Was a great talk. So I have two questions. One is, so from what I understood from your talk, you're talking about succinate being released by muscle cells and acting on cells bearing the succinate receptors, which are muscle cells again.
Yeah, they are muscle cells, but no muscle fibers. They are like stromal cells.
I see. The other question is, what is known about succinate as a modulator of epigenetics? Is there anything?
I don't know. Sorry, I don't really know.
So congratulations, Luis, beautiful work. One of the things about metabolites like succinate and especially lactate is that they can have secondary effects in whole organisms or cells, changing coenzymes, for example, like the NAD and ADH balance. And you did show some effects of lactate directly on an enzyme with the temperature denaturation and stuff like that, and that can't be because of NAD. But when you're adding them to cells or to organisms, how do you differentiate what's a direct effect of the metabolite versus its metabolic redox effects, for example? Or is it even important to differentiate these direct effects from the indirect effects?
Yeah, I mean, in the case of lactate, we did a lot of like NMR experiments to prove that lactate indeed binds to SAMP1. So again, it's very hard to show that. But of course, it might be like secondary effect. And we spent a lot of time like trying to figure out how lactate increases the thermal stability of UB2C until we come out with this hypothesis that it's actually facilitating the interaction between UB2C and the anaphase promoting complex by inhibiting SAMP1. So it's very challenging to do that. But of course, you can have secondary effects.
Great data, congrats.
Thank you.
And I'm curious about how did you find the lactate binding site in that protein? And else, which was the amino acid you mutated to prevent the lactate binding?
Yeah, I mean, we basically, we had like a structural biologist in our group that did all the modeling. I'm not going to go into the details on that because I really don't know. But he did a lot of in silico modeling and tests to come out with this. With this model here, and then we did all the mutations to test whether this was actually happening in real life. So we basically saw this model, and actually I didn't mention it here, but the SAMP1 is a zinc binding protein. So basically lactate stabilizes zinc binding to SAMP1 active site, because zinc has an inhibitory effect on SAMP1, but once lactate binds to SAMP1, it actually increases the inhibitory effect of zinc. But if you don't have zinc in the reaction, lactate has no effect. So it depends on zinc, and it's basically stabilizes a zinc binding to SAMP1 active site. And we did this mutation here.
Thank you very much. I'd like to invite. I would like to invite Dr. Alicia Kowalski. I would like to invite Dr. Alicia. She earned her medical degree and PhD in medical science from the University of Campinas and completed her postdoctoral training in Oregon Graduate Institute. She's currently a full professor in the Department of Biochemistry at the University of São Paulo and member of both São Paulo State Academy of Science and Brazil Academy of Science. Her research focus is on energy metabolism. Welcome.
Symposium. I don't think this is on, no. I can speak loud. I'm really sorry I wasn't here during the whole symposium. The reason is I'm capable of speaking loud because I've been teaching 16 hours a week. I would much rather have been here than in the classroom. But what I hope to tell you today and convince you today is that mitochondrial calcium transport is a central regulatory node in metabolism that's both sensitive to the levels of nutrients that an organism or a cell is exposed to and regulatory of the energy metabolism within that cell. So mitochondria take up calcium. They take up calcium through a mitochondrial calcium uniporter, which is in the inner mitochondrial membrane. And they take up this calcium from the cytosol into the matrix, down the membrane potential, which makes the matrix have a negative charge, which attracts this calcium. Mitochondria can take up calcium with much less affinity than the ER, but much, much higher capacity than the ER. So hundreds of times more calcium can fit into mitochondria than into the ER. Mitochondria can release calcium in exchange for sodium ions and also in exchange for protons. I'm not going to talk about this release. I'm going to talk about the NCLX, which is the calcium-sodium exchanger, and usually the main release pathway for calcium under physiological conditions. When mitochondria take up a lot of calcium and also are under conditions of redox imbalance, there is an oxidation of mitochondrial inner membrane proteins. This leads to inner membrane protein misfolding and the opening of a very large pore, which is called the mitochondrial permeability transition pore. This pore leads to the release of calcium and basically the release of any small molecule from mitochondria. It's incompatible with oxidative phosphorylation because of that, and it's typically associated with conditions of cell damage or death. Now, we found out that mitochondria were involved and were regulated by nutrient availability kind of serendipitously when studying our main and favorite model for obesity, which is the normal laboratory rat or mouse. So many people don't think about this, but our laboratory animals are reared under conditions of ad libitum feeding. And if you give ad libitum food to any animal, including ourselves, and nothing else to do, and don't tell them that obesity is not a good thing in your life, they're going to be obese. They're going to overeat and be obese. So you can see here in data from Mark Mattson's group, but we see the same both with rats and with mice, that young adult animals already have high body fat, insulin resistance, and inflammatory markers that are very typical of an obese human under ad libitum feeding conditions. And these conditions are not seen when you restrict the number of calories animals eat, and we typically restrict calories by 40%. And then you have an animal that looks more like a lean and healthy human. And of course, this also leads, and this has been known for almost 100 years now, this also leads to a very significant increase in life expectancy for these animals, this prevention of obesity through calorie restriction. Now, Inácio Amigo came to our lab, and he wanted to study the effects of calorie restriction on the brain, and specifically in excitotoxicity, which is a condition in which neurons die because of excessive activation through neurotransmitters that leads to too much calcium in the cell. And because of this, he decided to measure mitochondrial calcium uptake capacity because mitochondria are really important in buffering calcium within neurons under this condition. And what he discovered was that the change in the amount of calories that the animals eat changes mitochondrial calcium uptake. So what he did here was isolate mitochondria from the forebrains of animals that were either fed ad libitum and therefore were obese or calorically restricted in blue. And then he incubated these isolated mitochondria with a probe that fluoresces in green in the presence of calcium, but is not capable of entering mitochondria. It doesn't cross membranes. So it's only measuring calcium outside mitochondria. And then we give boluses of calcium and watch as mitochondria take up this calcium until they kind of give up and no longer take up calcium. And from that, we can calculate two different values, which are the calcium retention capacity, which is the total amount of calcium these mitochondria can take up, and the calcium uptake rate, which is the speed in which mitochondrial calcium uptake happens after the first addition of calcium. And very consistently, we saw that the calcium retention capacity for the same amount of mitochondria was higher in the mitochondria isolated from the lean animals, from the calorie-restricted animals, and the calcium uptake rate was very much faster in the mitochondria isolated from the calorically restricted animals. This difference was because of the opening of that very large mitochondrial permeability transition pore. And we know this because if we inhibit the permeability transition pore with cyclosporin A, we have equal calcium retention capacity and calcium uptake rates in both mitochondria from these animals. So this was the first evidence that we ever saw in the literature that changes in nutrient availability in an animal, a dietary change in an animal, changed properties of mitochondrial calcium uptake. And this to me seemed really important because we're putting together two very important regulatory nodes of metabolism. One is nutrient availability, and the other is calcium that regulates just about everything in metabolism, as we know, we that teach a lot of metabolism to undergrads. So we decided to look at this with different diets and in different tissues. And I'm not going to show you all the data. We published a number of papers on this over the years. But basically, this is data from Sergio Menezes' thesis, in which he also sees a change in calcium uptake rate and a change in calcium uptake capacity in the liver mitochondria of animals that were either fed ad libitum or calorically restricted. So many different changes in caloric intake, many different dietary interventions change mitochondrial calcium uptake properties in different tissues in different manners. And why is that important? That's important because calcium has the potential to regulate oxidative phosphorylation in mitochondria. There are a number of enzymes in the Krebs cycle and fatty acid oxidation and components of the mitochondrial electron transport chain that are regulated by calcium, and calcium inside mitochondria are where these enzymes are. So we wanted to see the impact of changes in calcium in mitochondria on oxidative phosphorylation. And this was the work of the postdoc of Eloisa Villas-Boas in the lab, in which she used modern techniques to quantitatively look at the impact of calcium on oxidative phosphorylation in mitochondria. And she has a large amount of data, but basically what we described was a Goldilocks effect of calcium on oxidative phosphorylation. Goldilocks effect means that neither too much calcium is good, nor too little calcium is good. There's a just right level of calcium that optimizes oxidative phosphorylation. And you can see here an example, state 3 respiration, state 3 oxygen consumption rates are oxygen consumption rates done in the presence of oxidative phosphorylation. And we can see that oxidative phosphorylation is low with low or high calcium quantities in mitochondria, but it is optimized in a medium concentration of calcium within mitochondria. And we described how this changes with different substrates under different conditions, but there's always a Goldilocks effect. Now, this is with isolated liver mitochondria. What happens in cells? So if you look at hepatocytes, you can follow the quantity of calcium in mitochondria using mitogecko systems, a protein that's sensitive to calcium within mitochondria within the cell. And you can change the amount of calcium within mitochondria through different interventions. For example, you can release calcium from the ER, which increases calcium in mitochondria, or you can remove calcium from the cytosol using a chelator such as BAPTA-AM. You can also silence either the mitochondrial calcium uniporter, so no calcium goes in, or silence NCLX, so calcium can't leave mitochondria. And using all these different interventions, we looked at oxygen consumption rates, both under basal and maximal conditions. And very consistently, we saw that whatever we did to change mitochondrial calcium decreased maximal respiratory rates in mitochondria. What that means is that mitochondria under normal basal conditions are at their Goldilocks level of calcium. And if you either increase or decrease this calcium, the capacity to transport electrons goes down. And I find this quite interesting because you would think, oh, cells are always operating under optimal conditions, but that's not really true. Cells don't use their total respiration, for example, under basal conditions. There is a reserve capacity. And I would expect cells to use mitochondrial calcium increases to increase their capacity under signaling conditions. But that's not what we see. We see that mitochondria have the ideal amount of calcium under basal normal conditions. They operate under this Goldilocks range of mitochondrial calcium. So I showed you that changes in caloric intake change mitochondrial calcium and that has an impact on oxidative phosphorylation. Now, as we were uncovering these effects, a lot of groups were looking at changes in mitochondrial dynamics and morphology and how this was impacted by changes in nutrient availability and also how this changes mitochondrial oxidative phosphorylation. So mitochondria are not organelles that are always the same in their size and shape. They're continuously changing size and shape, undergoing fission, becoming too small mitochondria, or undergoing fusion and becoming very large mitochondria. And these changes occur in response to changes in nutrient availability, and they also regulate oxidative phosphorylation. And we wanted to understand how these changes in mitochondrial morphology could relate to mitochondrial calcium. So to do this, we collaborated with Orion Sherahai. I actually went to his lab and did experiments. Full professors are capable of doing experiments. That was one of the things we proved. And what we did was generate cells with 3 different sizes of mitochondria. So they had normal size mitochondria, in which we didn't change their normal mitochondrial morphology. They had very elongated mitochondria, which Orion calls spaghetti mitochondria, or very small mitochondria, which Orion calls meatball mitochondria. He likes his food. So we were changing the shape of mitochondria, and we quantified this here. But very importantly, we were not changing the amount of mitochondrial mass per cell. So the area that mitochondria occupied, or the mass of mitochondria per cell, was exactly the same. And then we compared the calcium uptake properties of these different shaped mitochondria. And to do this, we got the cells and permeabilized the plasma membrane using specific detergents for cholesterol, which is not present in mitochondria. And then we did the exact same experiments I showed before, looking at mitochondrial calcium uptake rates and capacity. And very consistently, we saw that control mitochondria could take up a certain amount of calcium. Very elongated mitochondria could take up more calcium, and the very small ball-shaped mitochondria could take up very little calcium. And the shape also affected the speed, so the very elongated mitochondria could take up calcium faster, while the very small ball-shaped mitochondria could take up very little calcium. So the answer is yes, mitochondrial morphology does change mitochondrial calcium uptake. Now, I already showed you the effects of calcium uptake on oxidative phosphorylation and how this affects mitochondrial functions. What we wanted to know also was if there was an effect of these changes in mitochondrial calcium uptake on calcium in the cell, on cytosolic calcium. So what we did was mark the calcium levels within the cell with Fura2, which is a fluorophore that's accumulated in the cytosol and detects calcium in the cytosol. Then we depleted these cells of calcium and added calcium back to the extracellular media, this is intact cells now, and looked at calcium uptake back into these cells. This is a process known as store-operated calcium entry and is very important for the replenishment of calcium in cells after there's a lot of calcium signaling. Very consistently, what we saw was that store-operated calcium entry occurred in all cells, but the cells with average size mitochondria had average rates of store-operated calcium entry. The cells with very large mitochondria had very fast recovery of these calcium levels, while the cells with very small mitochondria had very slow recovery of calcium levels. And in fact, they never reached calcium levels that were equal to the control cells. And these cells with very fragmented mitochondria actually had ER stress because of lack of calcium in the ER. This means that changes in mitochondrial morphology change calcium uptake in mitochondria, but also have an impact on calcium uptake into the cell, changing cytosolic and ER calcium levels. So we saw that there's an interrelationship between changes in mitochondrial morphology and changes in calcium within mitochondria. And we also saw that changes in mitochondrial morphology and calcium uptake have an impact on cytosolic and ER calcium levels. Now, cytosolic and ER calcium levels regulate just about everything in the cell. So we've been having a lot of fun lately in the lab looking at how changes in mitochondrial calcium uptake impact on many different processes that we know are calcium dependent, but nobody looked if they change with changes in mitochondrial calcium uptake. And the first of these stories that I want to quickly show you is how changes in mitochondrial calcium uptake and changes in calcium in the cell impact on autophagy. So autophagy is a really important process in response to changes in nutrient availability. Autophagy is also important to recycle components in the cell once these components are damaged in some kind of way. Basically what happens is that LC32 locates in autophagosomes, and these autophagosomes fuse with lysosomes, which then degrade the cargo of this whole system, releasing nutrients that the cell can use under conditions of poor nutrient availability, for example. Many steps of the autophagic process have already been described to be regulated by calcium, so we decided to look if these steps were also regulated by changes in mitochondrial calcium uptake. And this was the idea of a student in the lab of Vitor Ramos who did his PhD on this subject, and a really nice work also. And basically what he found is that NCLX, the mitochondrial sodium-calcium exchanger, the pathway in which calcium leaves mitochondria, is a really important regulator of autophagy. And I don't have time to show you all the data. He actually showed this in many different manners. This has been published. If you want to read it, it was published last year. But I'm going to show you one of the indications of this, which is the system in which you're expressing in the cell linked to LC3 a GFP and an RFP. So whenever autophagy is initiated, you're going to have yellow fluorescence. And then the GFP is actually killed by the change in pH that happens when lysosomes fuse with the autophagosome. So you can see the fusion with the autophagosome by the appearance of only red fluorescence. And basically he saw that there was no change in these processes when we silenced the MCU, the mitochondrial calcium uptake pathway, but there was a decrease in both the initiation of autophagy and the fusion of the lysosome with the autophagosome when we inhibited the NCLX, both genetically and pharmacologically. So NCLX is a really important regulator of autophagy. And what he saw is that it's an important regulator of autophagy because of the effects of NCLX on calcium in the cytosol. Now, interestingly, not only does NCLX regulate autophagy, but conditions that regulate autophagy regulate NCLX. So if we go back to calorically restricted animals, and these are the livers of calorically restricted animals, and compare them to controls, it's well known that lower nutrient availability, such as calorie restriction, increases autophagy. And what we find is that calorically restricted animals also have an increase in the two isoforms of NCLX, the 100 and the 50 kilodalton form that we detect of NCLX. And this very consistently we find in different tissues of these animals also. So NCLX is a regulator of autophagy, but it's also regulated by conditions that regulate autophagy, which kind of makes it full circle. Now, NCLX is not only a regulator of autophagy, we also find that NCLX can regulate cognitive function. And this is really nice work done by João Vitor Cabral Costa, who was also a PhD student in the lab. So João Vitor, like many of my students who left the lab recently, was one of the students who spent a lot of time at home during the pandemic, which lasted forever for us here in Brazil. And he did a lot of in silico analysis because of that. And he was looking at NCLX and its expression in different tissues, and found that NCLX is very highly expressed in astrocytes versus neurons. And it's expressed in different parts of the brain in very high quantities, and also overexpressed relative to complexes of the respiratory chain, so this isn't because there are more mitochondria, and even relative to other components of the mitochondrial calcium transport machinery. So NCLX really seems to be a really important protein in the mitochondria of astrocytes. So what does it do in astrocytes? So we both inhibited NCLX pharmacologically in astrocytes or knocked it out, and we could not see changes in oxidative phosphorylation. There wasn't a change in the level of calcium in these mitochondria to a point in which you left the Goldilocks range of calcium regulation of oxidative phosphorylation. But what we very consistently saw when we inhibited NCLX was an increase in glycolysis to lactate production and an increase in lactate secretion from these astrocytes. Now, lactate secretion from astrocytes is very important because lactate generated in astrocytes is taken up by neurons and used as one of the primary substrates of neurons in cells. So lactate can actually make neurons much more functional. So we decided to look at what happened when you made your astrocytes generate more lactate in vivo in terms of cognitive effects in animals. So this was done with a collaboration with Juan Pedro Bolaños in Salamanca, and they basically knocked out NCLX either in neurons on the green data here or in astrocytes on the red data here, and looked at different cognitive tests to see how neurological function was. And very consistently, novel object recognition by these animals was improved in the animals that had NCLX knockout in the astrocytes, while it was decreased in the animals that had NCLX knockout in the neurons. And this is consistent with data showing that changes in lactate release from astrocytes regulate novel object recognition in other models in which there's lack of lactate release from astrocytes and there's a loss of novel object recognition in these animals. So I hope I convinced you here today that changes in caloric intake can change calcium transport in mitochondria, that this changes oxidative phosphorylation, that there's an interplay between the changes in calcium and changes in mitochondrial morphology, and that both of these can change cytosolic and ER calcium levels. We also showed that NCLX, the release pathway of calcium from mitochondria, is a strong regulator of autophagy, and that NCLX regulates lactate secretion from astrocytes, leading to changes and a positive change in cognitive function. Before I finish, I'm on a campaign as a bioenergeticist, trying to tell people that measuring oxidative phosphorylation can be done in many different ways, but perhaps the worst way to measure oxidative phosphorylation is to measure mitochondrial intermembrane potentials in mitochondria. I know it's very attractive because lots of companies sell probes and you can just put them in a cytometer. But when you're measuring the difference of concentration of something from one side of the other, such as the difference in concentration of cars from one side of the other of the avenue, you don't know what's going on. It could simply be that there's an accident over here and these cars aren't going forward. On the other hand, when you measure fluxes, when you measure oxygen consumption, how many cars go per minute, you're measuring real changes in oxidative phosphorylation. So Fernando Abdulkader and I, we wrote a review about that, and I also recorded a YouTube class about that if you want to see it. It's really something that has impacted on the field, I think, and there's a lot of data that's very confusing to people because these measurements are kind of hard to interpret. And finally, I have to thank everybody in my lab. I thank the former students who did the work that I showed you here already. We have current students with really exciting projects going on, and especially I have to thank our lab technician who is wonderful. So thank you.
Thank you very much. We have a few time for questions.
Well, amazing as always. So pardon my ignorance because of course I don't work with the transporters, I work with something else, but oh, actually it's a kind of transport, but let's leave it to later. Do you have enrichment of these transporters in contact sites of mitochondrial membranes? And do their amount or function depend on phospholipid or any other component of membrane?
Yeah, so the regulatory process and the properties of these different transporters are being studied. Lipids do interfere with them. There are subunits of the MCU that interfere with them. And we basically have seen that the MICU subunits are the subunits that change in response to dietary interventions, and they're the ones that change the calcium uptake properties. The changes aren't all or nothing. There isn't one subunit that's activating and one that's inhibiting. It's actually the proportion between them, so it's a little bit more complex. The regulation of the NCLX is much less known. Even its location in the mitochondrial inner membrane is now being questioned, if it's in towards the outer membrane or if it's on Christi specifically. There's an interrelationship with sodium transport, which happens in complex 1 to come in, because sodium has to come in so calcium can go out. So there are all sorts of questions on what regulates it besides the quantity of NCLX, which we do see change as with diets.
Because I noticed that you normalize by number of density of mitochondria and the size of the mitochondria, but not crystal density, right?
Yeah. We didn't quantify crista density. That's kind of hard. But the respiratory chain components are in the crista, so it's kind of a normalization to that.
Hi, wonderful talk. I have a question, but first I will tell you something which you may not know. So I just realized, your student Vitor probably worked in Newcastle.
Yes, he did with Victor Coralta.
Yeah, and Victor is a very good friend of mine, and we have many collaborative projects together. And your student Victor actually worked for my projectors too. He generated some data. And so that's a secret.
It's not a secret. I know. There's other stuff that he has still in his.
Yeah. So the question I have is, well, wonderful work, and mitochondria have many contact sites, such as with the ER. For example, there's something called MAM, mitochondria-associated ER membrane. And I'm just wondering, what's your opinion about how MAM regulates this mitochondrial calcium and so on?
Yeah, so that was a big question, especially because we were changing mitochondrial morphology by knocking down mitofuscin 2, which is the docking protein to the ER. So we were also changing the interrelationship with the ER. Now, our experiments with permeabilized cells, basically there's no effect of the ER in that uptake, because the ER uptake is less than 1% of what's taken up by mitochondria. So that interrelationship between the ER and mitochondria is not affecting those measurements. However, the changes in this relationship in the intact cells are certainly changing ER calcium levels. However, we also have an effect of knocking down the protein that leads to fusion, right? DRP1. That means that it really does seem to be more morphology than the proteins themselves. And the reason for that is not because of changes in inner membrane potentials or anything like that. It simply seems to be because of the change of the matrix size. And that's another thing that, I don't know if you know this, but there's data from Israel Seckler's group showing that store-operated calcium entry depends on NCLX. And basically, calcium has to come from outside the cell, go into mitochondria, leave mitochondria, and then go into the cytosol and the ER. I have no idea why, but our data really do confirm that mitochondrial calcium uptake has an impact on ER and cytosolic calcium. So it really seems to be true. Now, the function of this mitochondrial calcium uptake before it goes into the ER, I really don't know. Yeah, I think that has an impact on also sublocation of the mitochondria. I think the mitochondria that are really important here are the ones that are right under the plasma membrane between it and the ER. But we have not done the imaging to show that yet.
A quick question, a beautiful work, Alicia, as always. The impact of the activity of this transporter on the proton gradient to synthesize ATP.
Yeah, so when you're taking up calcium in quantities, this is going to decrease the membrane potential. It's a temporary decrease in membrane potential, which then recovers in mitochondria unless you have opening of the permeability transition pore. So the impact is going to exist in terms of a decrease in membrane potential only if there's a lot of calcium cycling, which isn't super physiological because typically mitochondrial microenvironments will have less calcium than a lot of cycling would allow for. But that hasn't really been quantified, and because it is very difficult to quantify membrane potentials in situ. in terms of millivolts because of all the problems with membrane potential measurements also. So I think that's something that we should look at, the impact of that.
Nice. Nice talk, Alicia. First, just a comment. Are you praying for Brazilian size? You seem to be praying for.
Oh, because I'm kneeling, yeah.
No, just kidding.
Yeah.
No, just a question. How could, in which step of the autophagosome formation, calcium act?
Yeah, it doesn't seem to be a single step, and that's one of the problems we have, because there's so many calcium effects in autophagy. We seem to see more than one effect, but it does definitely affect the initiation process. It affects many different steps, and this was characterized in the paper. It's actually one of reviewer comments and all that. It doesn't seem to be one single mechanism.
Hi, Alicia. Thank you for your presentation. I have one question because I usually use the mitochondrial membrane potential probes for evaluating the mitochondrial dysfunction as a starting point to investigate if some selected compounds are toxic or not for the cells, basically seeing the mitochondrial potential. And you said that we need to be careful about how and when we use this probe as an indicator of damage for mitochondria. Can you explain a little bit more why you have this opinion about these probes?
Well, there are many reasons, and I think the paper might help you most there. First of all, sensitivity. So you're not going to have much of a change in membrane potential when you have a very large change in oxygen consumption rates, because the change in the amount of probe accumulated at high membrane potentials is very small, and it's not a rate measurement. So first of all, you're losing sensitivity because of your method. The second problem is that the accumulation of these probes and the fluorescence of these probes is affected by many things that are not the mitochondrial intermembrane potential. And that's one of the main problems if you use this technique in isolation, if you're not using other techniques, if you're not controlling for all these other things. So I think there's a lot of advertisement that this is good. I think a lot of people are using this in an innocent manner, that they don't understand all these other effects that may happen. But truthfully, you probably could do a better screening with a change in oxygen consumption rates, which would be more sensitive, even though it is more work because you can't just use a normal plate reader. And I understand that problem, but you have so many less artifacts. Well, we could talk about what mitochondrial dysfunction is also. That's another whole talk. So what is mitochondrial function, and therefore what is mitochondrial dysfunction, I think it depends on what you're doing. It really does.
Thank you very much. And now another coffee break.
afternoon. So now we're ready to go to our last speaker. So Professor Marilia Silander is a chief investigator on the Cancer Metabolism Research Group in the Department of Surgery of Faculdade de Medicina da USP. She's the coordinator of LIM 26. She had an undergraduate degree of biology at USP, master's and PhD on physiology also at USP. And she was a research associate at the University of Oxford, Barcelona, and Potsdam. She had her livre docência, her professorship in 2004 at USP, and she's a board director of the Cancer Cachexia Society. And I have to add here that she's the ombudsperson for USP, so which means that the time she has to dedicate to research is less, so she has to do extraordinary work to come and show us.
Thank you so much. Thank you so much, Tessio, for these kind words. And indeed, I think the Ombuds office is quite a challenge, but we are coping so far, kind of. But it's true. It's really taking a token on our science, and I hope to present to you some ideas and data and that will incite more people to work with cachexia and understand it. So thank you again for the invitation. It's a very kind invitation. I'm very excited to be here at this wonderful meeting with distinguished colleagues and visitors from abroad. And I'll be talking about cancer cachexia. So Julio has spoken about it yesterday. But I really want to offer you the view that cancer cachexia, which is now considered a comorbidity of cancer, is not, let's say, as uncommon as you would think. And also, cancer is not just the tumor, right? Cancer is always a systemic disease. Cachexia is just an aggravation of the systemic effects of cancer. And unfortunately, not many people, especially at a clinical scenario setting, they take these into consideration. And we'll see that it really implies in worsened scenario, worsened survival for patients. So cachexia is much more than loss of weight or muscle mass, right? Although when we see a picture like that, of course it impacts us because it's really striking and it really calls your attention. But of course this is the end step in the disease, in the flux of the disease, which is actually an inflammatory syndrome, not really a wasting syndrome. Wasting is just a reflex of this inflammatory syndrome. And although it's not restricted to cancer, as Julio mentioned yesterday, patients with COPD, heart disease, and renal disease, for instance, do present cachexia as well, as among many others. In cancer, it is associated preferably to certain types of tumors, such as pancreatic, liver, colorectal, and lung cancers. And as you can see here, the presence of cachexia really impacts survival. That's 5-year survival. And already when looking at 6 months survival of the patient, there is a major and marked impact of cachexia. So as a syndrome, it is difficult to diagnose this, and unfortunately, not many people, as I was saying before, treat this in the clinical setting. But how can we diagnose cachexia? Let's talk a little bit more about this, but it should be diagnosed, nevertheless, in all the patients that come to the hospital with cancer or receive a cancer diagnosis. Because we have at least 50% of patients with cancer showing the syndrome, and 80% of all patients with advanced form of the disease will have it. So it's really prevalent in cancer patients. It impacts morbidity and mortality, as we said before,
And very importantly, it impairs the effects of chemo and radiotherapy while at the same time increasing the side effects, the undesirable side effects. It has it causes a major role in the quality of life, and it's per se responsible for 20 to 40% of all cancer-related deaths. So it's really a major thing in cancer treatment. And just for you to have an idea of the costs involved with the treatment or the non-treatment of cachexia, at this year, 2025, we have around $3 million per year being wasted or employed and wasted with cachexia, and that cost will shall increase to 4 million in 10 years as is expected. And as a syndrome, as I say, and it's difficult to diagnose a syndrome, it does present many different features. First is caloric intake. So most of the cachectic patients will show anorexia. That's not really 100% of cancer patients showing this symptom, but let's say at least 70 to 80%. But this is not the cause of cachexia, because when you think of it, the energy demand of a fetus, of a growing fetus growing in a pregnant mother, are much higher than those of a tumor that is growing in a patient. So that's not really the major thing in cachexia, has been thought for many, many years. And another argument to counteract or that poses another argument that shows that cachexia is not really bad nutrition, malnutrition, is that no nutritional therapy has so far been able to counteract the symptoms. Actually, no therapy at all can counteract cachexia. Nobody knows how to really suppress all the symptoms of cancer cachexia to the present day. Muscle function and muscle mass loss have been widely studied because, of course, there's an impact when you look at a patient with like the one in the picture that we saw before. But we can say that the muscle, although being very affected and whose function is very compromised by cachexia, is not really contributing to cachexia. It's much rather a victim of cachexia than actually a player or a trigger of cachexia. There are major neuroendocrine changes, and we will speak a little bit about catabolism. So this is a feature that I want to put forward here as one of the major problems which cannot be really counteracted efficiently with 1 or 2 or 3 types of therapy at the same time. And pharmacological therapy or non-pharmacological therapy have so far failed to suppress the symptom. And what is this metabolic chaos that I'm showing here? Actually, even if you increase caloric intake, if you give the patient a lot of calories, the patient will not regain health, will not decrease the symptoms. And why is that? It's because in cachexia, all the compartments, all the organs, all the systems of the body are rendered incapable of utilizing energetic substrates. So it's a compromising of the capacity of metabolizing substrates, not really the lack of substrates. That's why, of course, nutritional therapy also fails. And we will talk a little bit about that, especially in regard to the liver. And what is causing that? As I said to you in the beginning, cachexia is now envisaged to be an inflammatory syndrome. And central inflammation, so there is brain, central nervous system inflammation, and peripheral inflammation, chronic systemic inflammation, are driving these changes that cause cachexia. Perhaps not another, but the most important question here is why patients with the same tumors, the same diagnosis, and the same stage in tumors disease at the same stage will or will not present cachexia. So I may have one tumor and my friend may have the same tumor at the same stage, and yet I will develop cachexia and my friend will not. So what is going on here, right? That's a very important question. And part of the answer to that is that it's not only the tumor that is present, but also how the organism as a whole responds to the presence of the tumor. And this conjunction of responses will cause cachexia or not. So as we were saying before, inflammation is a very strong component of the syndrome. And we have both the tumor and the host secreting different factors, inflammatory factors, that will have an impact in all the organs and tissues of the person. So the tumor, for instance, secretes lipid lipolysis-inducing factor, many other cytokines that are not even represented here, while the host, for instance, will show increased CRP, and that's a way to measure cachexia in the presence of the tumor, and many cytokines, as well as like eicosanoids and other types of factors, are secreted by both components, the tumor and the host organism. And this joint action of all these factors will elicit changes in the adipose tissue. That's my favorite. I always study the adipose tissue, actually for 30 years, but today I will not speak about adipose tissue. Rather, I will dare to speak about liver and muscle in cancer cachexia. And you can see in this picture that there are many changes that are caused by the disease, and we shall comment on them briefly because we do not have much time. Well, if the two components are important, if the tumor is important and the tumor host's response is important, let's have a look first at the tumor, right? What is going on? So cachexia is a continuum. It starts with precachexia, where when you don't have that image you don't have the same, you don't have weight loss that is so comprehensive you can actually see like in the image that I showed to you. Then this develops into cachexia and then refractory cachexia, which corresponds to the image of the person that I showed to you. And of course, the tumor is also progressing during this span of time. But the tumor is not only composed, as you all know, by tumor cells, but also by many different infiltrating cells and not infiltrating cells that compound, that are part of the microenvironment of the tumor. And we have, and I'll go very briefly and very quickly about this, we have looked at this, and when I say we have, is Emílio Matos and Joana Lima, my former students. We have looked at the tumors of cachetic and stable cancer patients, and they had the same tumor at the same stage, and we were looking at the differences between these tumors in terms of components of the microenvironment of the tumor, and also in terms of the secretion that this tumor had. And just to give you some examples, this is cancer cachetic patient, this is stable cancer patients. We could find that there's a different relationship between M1 and M2 macrophages within the microenvironment of the tumor. So that's already one, let's say, very important feature that will obviously be linked to differences in secretion. And as we can see here, as an example, I bring the secretion of TNF-alpha, which is much higher in the cancer cachetic group, and also chemoattractants and many other cytokines. But this is published work, and it's not very new, so everyone can have access to that and have a look. Also, a little bit later, Gabriela, who's here, there she is, and she was a bit shy, but I wanted to show her. There's a picture of her there as well. She was supervising Ana Taranco, who was a mobility student, and Joana was a PhD student at the time. They looked at the tumor of this cancer cachetic or stable cancer patients, and they found that, for instance, myostatin secretion was increased in the tumors of the cachetic group. And why is this important? Because myostatin is very relevant for muscle biology. So it will, if there's a lot of myostatin, you will have hypertrophy. And the muscle of cachetic patients does not present any changes in the secretion or expression of this factor. But actually the tumor is secreting an increased amount of, or increased concentration of this cytokine. And then following that, Joanna, and this is in published work still, had a look at the proteomic analysis of these tumors from cachetic and waist-stable cancer patients, having found many proteins that were differentially regulated in the two groups, from which two called our attention. One class of proteins was that of heat shock proteins, and the other one was hypoxia-inducible factors. So this was really interesting, and we thought, okay, so maybe the cachectic patient tumor is producing more HIF and is releasing that into the circulation, and that has an impact in target tissues. But does it also have an impact in the tumor, and why is this happening, right? So Renata de Castro, another PhD student, had a look at this hypothesis and what is causing this, what is the difference between the two groups of patient tumors. And what she proposed, that was her hypothesis and she wanted to test, was that there was a difference in autophagy in the tumor microenvironment. So normally what would you have was normal autophagy, and then delivery of antigens, and T cell C, CD8 positive cells would come to the site, and oxidative phosphorylation and fatty acid metabolism would increase, while glucose metabolism would be decreased, and that has an impact in inflammation. And there is no excessive inflammatory response from this tumor. However, in cachetic conditions, due to unbalanced autophagy and continuous presence of increased amounts of HIF, what would happen is that the infiltrating immune cells would pass away to cell heaven, and then we would have more damp, and then this would elicit the tumor as a whole to produce and secrete more cytokines, or more inflammatory cytokines, inducing cachexia. So this is a hypothesis. She has tested it, but the number of patients is not enough for us to really say that she could prove it. So it still remains a hypothesis. And an interesting idea is that already back at 2010, this group, Martinez, Outshorn, and colleagues have proposed that stromal autophagy in the tumor was occurring at a large extent as to feed cancer cells. It's a little bit teleological assumption, but anyway, it's interesting. So, okay, so now at least we knew that the tumor cells and the inflammatory cells of the tumor, and also we know that the inflammatory cells from the host, him or herself, are releasing cytokines when the tumor is there. It doesn't matter if the person is cachetic or not, because this is happening in both of them, just to a larger extent in the cachetic patient. And then we wanted to have a look at the liver to start with. Why? Because we said there is metabolic chaos, and this is a problem, this is a situation in which intermediary metabolism is really compromised. We know that. There's the metabolic chaos that we say. And the most important organ controlling metabolism is, of course, well, at least the central organ controlling the metabolism is the liver. And when you look at that, and this is old and or a little bit more recent work, we can see that in animal models, there is a diminished liver ATP content and diminished rate of ATP production by the organ as well. So this is Jurassic results. We were really looking at the liver back in the 90s and starting of the 2020, maybe 4 or 5. And what we did find at that time is that the liver was steatotic. It was really accumulating lipid droplets. And it was very strange because, you know, we expect the liver to use fat, not only it will store glycogen, but it uses lipids as the main energy substrate, because of course it will not use glucose. It has to cope with, it has to maintain glycemia, right? But if the content was much higher in the, tumor-bearing, this is rats, not mice, the lipid content was much higher in these cachetic rats, and also the liver was uptaking more fat at that time. So we thought, well, maybe this is strange. Maybe it's the mitochondria that are not really working very well. And some people had looked at the mitochondria before us, and what they saw is that, you know, the number of mitochondria and the mitochondria crista, and also the density and the area of these organelles was not changed in the animal models of cachexia. So, okay, so it's not the mitochondria number or density, so maybe it's function, right, that is changed. And the liver is doing extra work here in cancer because the tumor, as you know, will produce lactate, throw it in the circulation, the liver uptakes it and has to convert lactate to glucose de novo, and that has an energy impact. But if it's accumulating lipids, maybe it's not being able to drive, you know, its energies demands from lipids. So we thought, okay, let's look at the mitochondrial function to see whether lipids are being, can be oxidized in cachexia. And the first step, the controlling step of lipid oxidation is the entry of long-chain fatty acid into the mitochondrial matrix, right? And this is performed by a set of enzymes, carnitine palmitoyltransferase 1 and 2, which make it possible for long-chain fatty acids to transpose the mitochondrial membranes. When we look at that, again, Jurassic work, we saw that maximal activity, maximal catalytic activity of CPT1 and CPT2 were decreased in tumor-bearing animals as compared to control ones. And also, another impact of these was that there was a decreased content of ketone bodies in the circulation of these animals. So this is very bad, right? Because ketone bodies control proteolysis. They impede proteolysis in the skeletal muscle. And there's no ketone bodies, not enough ketone bodies. And I show this picture here just to show you how old I am, because we found an isoform of CPT2 that cannot be seen here, because this is so old that it could not be transposed to an electronic file. Okay. Another thing that we found at that time is that the disposition of CPT1 and CPT2 within the liver acinus, and which is an important thing to consider when we are thinking of metabolism, was changed by cachexia, as you can see here. So normally, pluripotent zone of the liver will be involved in the oxidative processes. And CPT2 here, as you can see, has moved preferentially to the perivenous zone, which cannot do these things, especially because oxygen doesn't reach this region. So what is causing that? And we first looked at it in animals when we found that, you know, time in a time-sense way or a time-increasing way, we had these animals showing more and more inflammatory cells in the parenchyma of the liver. And then we look at more recently at patients, and we found the same aspect in cancer cachetic patients. So there is clear infiltration of monocytes in the liver of these patients, and both the inflammasome pathways and the NF-kappa B beta pathways are increased in cancer cachetic patient liver, and also in animals. As you can see here, this is the work of Das Neves Rodrigo, who worked with us. Back to the Jurassic, we showed also that indomethacin treatment could prevent this effect of cachexia. So it's not only cytokines involved in that, but inflammation that also involves prostaglandins, prostaglandin E2 in special. So what about the mitochondria themselves? Okay, the function is different. The number may be not different, but other aspects of mitochondrial dynamics have been proposed to be changed. So this is not 2026, not a forward-thinking paper. It's 2016. And Jim Carson had proposed, and his colleagues proposed, that all these aspects would be changed, right? So biogenesis would be really impacted, dynamics would be altered, mitophagy would be really pumped up, and also apoptosis, and function would be decreased except for the production of reactive oxygen species. And then Rosa Caldwell in 2020 showed that it's not really everything that is expected that is happening. So I'll try to go fast because, of course, I'm speaking too much. But what she's shown is that, for instance, mitochondrial turnover related proteins are not really changed in the scenario of cachexia, at least in her model, which is the Lewis lung carcinoma. Fusion and fission are changed as also markers of autophagy, but in a time-wise way. So depending on the development of cachexia, you have different results, right? And when we transpose this, and then there is Gabriela again, when we transpose this to the human scenario, it's a little bit more difficult to think and consider because, of course, if we have a time-dependent change, how can we do that in a patient, right? We cannot collect many liver samples all along the progression of cachexia. We are very fortunate just to have a liver sample from now, once and only. But at least with this number of patients, Gabriela could not really find any difference, and that's fission with 2S, not 1S, I'm sorry about that. But she could not, with this number of samples, find anything. So if the liver is changing, its biology and its function, it's really having part of its, let's say, job compromised, that will affect, as we mentioned before, the muscle. But the muscle, as Julie was putting forward yesterday, is already very affected by cachexia. And I mean, mainly it's being wasted. There's increased proteolysis, decreased protein synthesis, as you can see here. And mitochondria are also affected, right? In terms of structure and capacity for oxidative phosphorylation, calcium handling, Alicia, everything is changed in cachexia. And when working with mice and doing some transcriptome analysis, Giquet and colleagues showed that most of the things mitochondria-oriented were changed by cachexia. So autophagy is increased, there's a depolarization of the mitochondria, there's swelling, as mentioned before, there's more permeability, there's less respiration, there's decreased transmembrane potential, the amount of mitochondria is changed, there's decreased fragmentation of mitochondria, decreased density also, and decreased consumption of oxygen. That's all in animals. But then we think of mice and men, and then we can think that maybe what applies to mice, and I'm sorry because most of us here have worked or work with animal models, not always, can be translated to humans. So this is an example. That's the proteasome. So proteolysis occurring in the muscle of cachetic mice have been extensively studied. And when looking at the genes that are expressed in people, in cancer patients, and in animal models, many differences have been found. So the so proteasome activity is not really the important thing in the patients. There's something else going on there. And then Gabriela again, so she's the star today, she should actually be speaking, has looked at the muscle of patients. She's published that already. And she's found that, firstly, the sarcomere disposition was disrupted in the cancer patients. There are figures of altered triad morphology, and there's the presence of swollen mitochondria, which is not unexpected, right? So she found that mitochondria area was increased despite no change in the number of mitochondria. And let me do the following. I'll talk about this and then go back. And looking at mitochondria-related proteins, mitochondrial biology-related proteins, she also found that FIS1, for instance, and mitofusin-2 were increased. So both fission and fusion were increased. This figure here, this picture, shows the mitochondria of the cachetic patients. And it's interesting to note that there's a lower electron density in these organelles that are depicted here. That means that these mitochondria are not so functional, right? And when looking at mitochondrial biogenesis, just like in the animal models, no changes could be seen. And another interesting aspect is that we could not really find inflammation in the muscle. So the muscle seems to be afflicted by inflammation, but already produce inflammation or spread inflammatory factors. And I'm finishing. Oh no, I'll go back to this one. I'm not finishing. Oh yeah, looking at different proteins in the autophagocytosis pathways. She also found that they were increased in the muscle of these patients. So LC3-2 and HCG5 and 7, for instance, that we have been listening about these 2 days. And also apoptosis factors were different, were increased in the muscle of the patients. So you can see here, for instance, apoptotic bodies, and also there's this, I think, heterochromatin here in this white arrow. I'm not sure you can see from such far. So with that, we can say that, okay, everything is a mess. The muscle is very affected. The liver is very affected. But, okay, that is a description of things. So does that help the patient anyhow, right? We can even know the mechanism, but we have to find a solution for that. So what we are now doing is that 2 other students of mine who are here, Gabriela, you see I'm obsessed, Giovanna and Marina, very bright students, are looking at on the crosstalk among tissues and the tumor in cachexia. So we believe that the muscle or the liver or whatever tissue you think is impacted by cachexia is so because of the constant communication, inflammatory communication that is originated in the different organs. So our idea is that most of this communication is performed by factors that are circulating within vesicles in the blood. And these vesicles are mostly, but not only, exosomes. So we're looking at exosomes, and now we are so lucky to have the tissues from these patients in surgery. So what we have now is that this grows, they take the sample, muscle sample, tumor sample, adipose tissue sample, not liver sample so far, but we expect that one day we'll be able to have that as well. And they make explants. So they work with explants. We incubate these explants. We isolate exosomes from these explants, and then we incubate the isolated exosomes with the target tissues to see what they elicit, in the hope to find that which component of the cargo of exosomes is eliciting the bad things that are occurring in each and every tissue. So thank you very much. That's my group, and that's all the collaborating groups in North America, Brazil, and Europe. And we are, of course, especially thankful for the funding, science funding agencies, and also for the hospitals that work with us, especially the surgeons who are very kind, and the patients who offer themselves to be analyzed by us. Thank you so much.
Thank you very much, Marilia. I think it was a very nice final talk, because it integrated many things that people talked about around the days in a disease-relevant system. Right. So we are now open for questions.
Thank you for that. I'm not going to ask about mitochondria. I'm curious about these livers with all these lipid droplets and basically the same amount of mitochondria, although morphology and location are also important. If there are changes in peroxisome activity, which is also so important for long-chain fatty acid metabolism.
Isolate some peroxisomes, because at first it was really like, it was an accident because we could not really isolate mitochondria.
Yeah, they come together, yeah.
But then we finally managed, and we did not find any differences in the number as available, but we did never test function, so I do not know. But nevertheless, when we decreased inflammation, we could restore the CBD activity, and we could restore lipid metabolism, because we also looked at VLDL secretion at a time, and it was really impaired, and the vesicles were much smaller, and the content was totally different. And by decreasing inflammation by many different strategies that we tried, we could restore normal function. So, I don't know, maybe peroxisomes are, well, didn't look, I'm sorry, I have to confess.
Thank you.
Navigants.
Hi, Marilia. Nice talking. I'm not talk about muscle. It was a nice talk. My question is, have you measured the lipogenesis in this liver? I know that's quite difficult, right? The fatty acid synthesis, perhaps it's making more fatty acids.
Well, because we were doing this with radioactively labeled palmitate and other fatty acids. We did not see that, we saw more esterification, but not really synthesis.
Synthesis, right. So, and what about the FFA flux to the liver? Perhaps it receives a lot of fatty acids. That's why they are accumulating a lot.
Yeah, we saw the uptake is increased, and
right.
Also the transporter, well, FAT, right?
Right.
It was increased, but not only in the liver, but also in the adipose tissue. So maybe it's a detoxing strategy, a desperate detoxing strategy. But the liver was accumulating that, and it was really impairing the function. One thing that was found by another group is that, because of course the lipid droplets can be adjacent to the mitochondria, right? And especially in the muscle, in the liver, not so clearly, but in the muscle, that's very important for muscle function. And this is decreased in cachetic liver and muscle. So there is also something going wrong in terms of this disposition, the topography of lipid droplets and the mitochondria.
Curious if I could just comment. Mitochondria around the lipid droplet typically are synthesizing mitochondria. They're making citrate for fatty acid synthesis and not degrading mitochondria. So it's weird that you have less, but you have more lipid droplets.
Yeah, and also the FABP, it's increased. So transport is increased, not only in terms of the amount of ABP, but also the topography, as I showed, the periportal and perivenous zones. It's much more evenly distributed than in the control animals. So, not secreting it very well, and it acts a little bit like a denervated liver. Because we tried denervation as well, and the results were a little bit like that. But it was just let's say, it was not a wonderful protocol. It was with phenol that we denervated the liver, but we could see very similar results. We published that a long time ago as well.
Any more?
Hi. So, like often, you have mitophagy and lipophagy and general macroautophagy. So there is a mitophagy reporter mice, developed by Ian Ganley in Dundee in the UK. And he has done very elegant study where he, induced physiological autophagy in the mice by starvation. And what he sees that not every tissue is inducing mitophagy. That means that even if you induce general macroautophagy, it may or may not activate selective autophagy of certain substrates, and it depends often on an organ-specific manner. So it is possible that in your system, macroautophagy could be affected. That is not, affecting mitophagy, but it might affect lipophagy. So it will be interesting to see. So if you maybe try to see localization with LAMP1 and LC3 with the lipid droplets or mitochondria, you can maybe able to distinguish between the two selective processes.
There's more work for Gabriela, but now she's a professor at the university, so she will have to do it with her own students. Thank you so much.
Thank you, Marilia. So if there aren't any more questions, we are going to have this final plenary discussion. So I'll ask that the speakers of the afternoon please come to the podium.
Eu já dei eu pisei na minha própria calça. Eu acho que é melhor que você naquele ou aqui, que tem mais gente. Obrigada. They're so shy.
So are there questions that anybody would like to pose to the speakers of the afternoon?
Professor Marilia, it's just about the curiosity. All the cachexia patients have like hypoxia inducible factor increased? Look, not all the cancer ones, just the patients with cachexia.
Yeah, we do see that within the tumor environment and also in the circulation, HIF, for instance, is increased. That's why Joanna went to Oxford to work with Peter Ratliff, because he, of course, is involved in the discovery of the control of this system of hypoxia. But it's increased. I mean, it's not that it's not present in the other patients. We have only analyzed, in these cohorts that we showed, we just analyzed colorectal cancer patients. So the one that really causes more cachexia is pancreatic cancer, and also lung cancer patients may also have problems. But in lung, you have like a dubious thing there. So in pancreatic cancer models, you also see that, but in patients that have never been shown before. So I really do not know if other tumors will elicit the same effect.
Do you think it could be because of the lipid accumulation in the liver changing the dynamics of oxygen getting to the tissue? Because the diffusion of oxygen in the liver is known to be bad to begin with. And the presence of these lipid droplets might
Thank you. I was wondering, sorry, also the lack of iron, because iron is necessary to degrade lithium alpha, because protease hydroxylases work with iron. So cachectic patients should have like less iron in circulation. So also stimulating the inhibition of degradation of lithium alpha as well. So
Very true. And actually they do have, so anemia is one of the hallmarks, and it's a criterion for diagnosis. And also in a Jurassic paper we have published something, this is a very funny story, but I will not tell it in public. We have found that many different ions were decreased in the circulation of cachectic animals and patients. So yeah, you have totally a point there.
Yeah, I have a naive question for Luis. I really appreciate your talk and what you've shown about the lactate and the succinate and overproduction of succinate that is exported out of the cell and that will bind a receptor. I was wondering if it's possible that there is also an accumulation of succinate into the cells, because it was not clear in my mind if this accumulation is possible or it's an overproduction and an excretion of the succinate.
Yeah, it's possible to happen. Like, for example, during ischemia, heart ischemia, for example, you have a huge accumulation of succinate in cardiomyocytes. But the problem is succinate functions as a reservoir of electrons. So when you put back oxygen, like you just produce a lot of like ROS, and it causes like myocardial infarction damage. So under hypoxia condition, you can have this accumulation of succinate inside the cells. Okay. But the heart can also secrete succinate as well.
More questions?
now to Professor Alicia, you mentioned about the calcium, like the calcium handling and the calcium techniques to evaluate the calcium inside of the cell and mitochondrial calcium handling. So could you comment more about the technique that you show in the slides?
The calcium uptake in
yes.
mitochondria. So one of the problems is that mitochondria take up so much calcium that calcium actually precipitates within mitochondria. And if you use a probe that's inside mitochondria, you're going to see the free calcium concentration, and you might underestimate what's in there because the precipitate is not going to be seen by your probes. So when we use like the protein probes that the encoded probes that we're using, we can underestimate calcium uptake. So this quantitative mechanism to see calcium uptake that either requires isolated mitochondria or permeabilized cells, we're looking at calcium outside and estimating how much is entering by what's disappearing from the media. It's just the technique that works for us to see this quantitative uptake. And we also have a published technique on how to measure NCLX activity, how much calcium is leaving mitochondria. Basically, we put calcium in mitochondria, inhibit the MCU, and then look at calcium efflux promoted by, for example, a loss of membrane potential or something, which is either sodium dependent or independent. So then we can measure the rates of these other proteins. And it's important because the amount of calcium that's taken up, the rate that's taken up, is not in correlation with the quantity of the proteins all the time. There's a lot of regulation that's not just protein quantification.
It's, again, maybe very ignorant.
Microphone here, I'm sorry.
And what about the intermembrane space? Does calcium accumulate there as well? And how do you tell one from another?
Well, you wouldn't expect calcium to accumulate in the intermembrane space. First of all, the outer membrane is quite permeable to calcium because of VDAC, of the main protein that allows most small molecules to pass through the outer membrane. The intermembrane space, it doesn't have the negative charge of the matrix that attracts the calcium. So calcium is really attracted towards the matrix, and it should accumulate in the matrix, and it usually accumulates and precipitates with phosphate, which is very abundant in mitochondria, because mitochondria use phosphate to make ATP. So you wouldn't expect it to be there. But there are people who have put these encoded probes in different parts of mitochondria and have seen that the concentration in the intermembrane space is pretty similar to the cytosol.
Okay, since there are no more questions, I understand that everybody has all the doubts are cleared. Nobody, so we have been very efficient in delivering. Right. Check. To check. Okay. So in this, just a minute. So this last minutes, I would like to thank again all the speakers for the very, very nice presentations and for the lively discussion that was here. I also thank you people who came to the meeting, because you also participated a lot. I hope this was I'm sure that this meeting was inspiring in more than one single way. Just both for sparkling new scientific questions, and as well as the entrepreneurship part that Ella covered so well. And now I'm going to give the floor to Sovan, and then
So I will say a lot of thanks, but first thanks to Luis for keeping the panel discussion alive. So tomorrow I'm visiting your lab, you're presenting, and I'll ask similar amount of questions. So just be prepared. So thanks to all the audience, and all the amazing speakers. It's great to see science, such amazing science in the Sao Paulo region and beyond. And great thanks to Edesio. So this conference is single-handedly done by Edesio and with support from USP. But I'm glad that the idea originated from our collaborative seed grant with Birmingham, and we have reapplied for this grant, and hopefully next year we can have a 3-day workshop if we get the grant renewed. And just some of you who missed some information yesterday, I'm from the University of Birmingham, and we have a Brazil Institute which offers a lot of funding opportunities, specifically with the state of Sao Paulo, also specifically with USP and Unicamp, for example. There are seed grants as well as visiting fellowship for researchers to go to Birmingham, which is fully funded by Birmingham, as well as there are other Birmingham FEPESP grants as well as UKRI, UK funding with FEPESP. So there are multiple, collaborative opportunities ranging from seed grants to visiting fellowships to larger external funding. And I'm happy if you have any questions, or if you want to engage with Birmingham or any researchers, please feel free to contact me. So over to Edesio, and thank you so much, and thanks to all the amazing speakers for accepting the invitation.
So I'd like to thank, at the end, I'd like to thank the people that made this meeting work. And I'm talking specifically about Andrea and about Eder. So I want to raise some applause for them. Oh, of course, and of course, Andressa here has been covering everything to go to the social media. Okay, thank you very much, and have a good Friday.
It's Thursday.
Obrigado.